Hello. Mistry, to please honor our guest, Professor Dr. Laszlos, and felicitate them with floral bouquet. Now I'll request Madam to felicitate Dr. Ravi Gupta. Now I request Professor Dr. Nitin Labane to come and introduce today's symposium to our participants, which is organized by Department of Botany, Powell's College.
Good morning, everyone. I hope you can hear me. Welcome to the International Symposium on Recent Advances in Plant Sciences. I know I will first like to say sorry to you all because this symposium has been envisaged in very short duration of notice. So we didn't get any time to communicate with not many people. But then my professor was here, Professor Lazar Shabados. And since he's in India, then what could be a much better way to welcome your professor rather than organizing an international symposium in his honor. So this international symposium is basically to honor my professor, Professor Lazar Shabados. He's there with us. He has been my mentor all the time. Then a very good friend of mine, Dr. Ravi Gupta, he also consented to be a part of this symposium. And it's an honor, Dr. Ravi Gupta, for you being here with us, all of us. So then the things started to fall in place. Then I called upon another friend of mine from Spain. He was there with me when I was doing my postdoc in Hungary. So he also consented that, okay, he will be there for the session, but he requested that he will be there online because we are not in a position to pay his TA and DA from Spain. So he will be there with us online. So the basic theme of this particular conference is that being students of botany, we hardly know that where the path will lead us. And what kind of research is going on across the globe is very difficult to understand. Even if I tell them, then they will say that, okay, fine, you are doing it, then what's the big deal? So, so I thought that why not to hear it from the speakers themselves who has made the name in the field of plant biology. So with this particular theme only, this particular international symposium has been envisaged. So once again, I thank all the guests who has come from India and abroad. I also thank all the participants who had been here for this particular symposium. And I assure you that by the end of this symposium, you will get to know what research, what level of research is going on across the globe and where we need to go ahead from here. Thank you very much. Now I request Asmita Adam, the Department of yes, yes. Yeah. The tradition, since we are coming here for symposium, so what we do normally is that we water the plant and we inaugurate that particular function. Now I request all the guests to be there. The plant is kept out there, hibiscus, which everybody knows it. So we will water the plant, and that will signifies that the international symposium has been inaugurated. So I request the guests to please be there and water the plant and declare that the symposium is Ravi Gupta. She is also faculty in University of Delhi.
To please honor our guest, Professor Dr. Kailash, and felicitate him with floral bouquet.
Sorry.
Felicitate Dr. Ravi Gupta. Yes, Professor Dr. Nitin Labane to come and
Yes, Ravi. It's a check. Okay. Hello. Now I request, since the session has been inaugurated, now I request the Asmita Madam, the head of Department of Botany, to tell us regarding what botany department is to all the participants. Thank you.
Thank you, Nitin sir. A very good morning to one and all. The guest on the dais, Madam Gauri sir, and my all dear students. Now I am here to speak about the department. Botany department, one of the oldest department in the Pavans College, established in 1946. It is the department which deals with the study of plant sciences. The subject which is changing according to the climate also, and relevant changes are there. It is the basic sciences, which is very much necessary. Now the department has a tissue culture lab. It has got a mycorrhizal lab. Labs are there. Also, the faculties, which are eminent one in each and every, what you can say, the core. Like some are being specialized in molecular biology, some are being specialized in mycorrhiza. Some recent ones who had joined the department. Those are the youngsters who also have a research interest in nanotechnology and also microbiological aspect. The department also has the students who are excellent in research also. Most of them have presented papers in conferences, and also they have 1 or 2 papers published in an international journals. There are few students who have qualified competitive exams like NET and SET also. The guidance by the staff members and also the efforts of the students. With this short introduction, I conclude and also request now for Vishal sir to speak about the Bhavan's Botanical Society. Vishal sir.
So very good morning to all of you. Respected guest, Professor Laszlo. Invited guest, Dr. Ravi Gupta. I think here Mrs. Gupta is also there. Invited participants, no doubt. I first of all welcome you all. No doubt, this symposium is organized by Department of Botany under the aegis of Bhavan's Botanical Society. Being a secretary of Bhavan's Botanical Society, it's my important role to explain about what is exactly Bhavan's Botanical Society has done since from the time of its inception. Most of the youngsters are not aware about the Bhavan's Botanical Society when they enter it first time in the campus. But in due course of time, they realize the role, significant role, what is performed by this society. Dear friends, this society has been established in year 2005. And in same year, the very first seminar which was organized, it's almost 20 years back now, almost. That was on the intellectual property right. Now everyone it is knowing about it today. But I'm talking about the 18 to 19 years back. So that was the beginning. So it was just a crawling baby, 18 to 19 years back. And day by day, we started to give some new initiatives. And every year, at the seminars, workshops, or conferences, they were conducted in the interest of students of botany, that is plant sciences. The researchers, they were invited over here. Various scientists, in due course of time, they joined us. And we achieved the important, because we have done very, very good important achievement in due course of time. That was nothing but our profile was recognized at national and international level by various peoples. No doubt this society was established to inculcate the interest among the plant sciences, among the students basically. And as a result of it, various students, they started to study plant sciences from different perspectives. As Madam and Nitin sir has told that, this department has established very good laboratories as well as the facilities for the students to study plant sciences. Most of them, they have achieved their role in such a way that their contribution has been get recognized in the form of research publications as well as in the form of the competitive exams what they have so far cracked. I think Nitin sir has told that this is the one of the you can say one of the most importantly recognized department in the Maharashtra, perhaps, who has given maximum number of set or nets qualified students as well as the get qualified students CSR. So this is simply possible because of Department of Botany and Bones Botanical Society. They have organized variety of workshops, variety of seminars for these students, and we are nowadays getting the fruits of this. So dear students, as an initiative of the society's function, we have organized this international symposium. I welcome all of you on behalf of Department of Botany as well as Bones Botanical Society. And today's symposium, it will give you definitely fruitful discussions. So once again, welcome you and thank you very much for your participation. Nitin sir, I think we have missing something that Mrs. Dr. Gupta is also over there. So I request you to felicitate our token of love. I'm sorry, madam, we have forgotten about in this particular course. I request Asmita madam also to felicitate. I think Dr. Sachi is also over there. Nitin sir. Dr. Sachi, I think you're there. Please. Asmita madam, please offer bouquet to Dr. Sachi. She has been there with us also. Please come. Here is Dr. Gore sir is also here, so we welcome him. Asmita madam. Thank you, sir. Now I hand over this mic to the Sharayu.
Now I'll request all the dignitaries to kindly take their seats in the front. And there are, yeah. I'd like all the participants to kindly note a change in the schedule. The first lecture that we are going to schedule is of Professor Dr. Laszlo Semedos, followed by lecture will be given by Dr. Abhi Gupta, and then finally we are going to have an online lecture which will be conducted by Dr. Riaz. So now I will request Dr. Laszlo Semedos to please. Yes. Just yes, yes.
Is there? Which PPT? Which PPT? Last one.
So within second. Yes. Thank you. So. I'll request Dr. Nitin Labane to please come on the stage and introduce Professor Dr. Laszlo Severos to the participants.
Thank you, Sharil. I know Laszlo from last 10 years or so. Okay, but then you all should know it. The person who is sitting out here, we all know that who is he. He did his PhD from Hungary. After that, he did his postdoc from CIAT, that is International Center for Tropical Agriculture, from Colombia. So the Colombia, when you're talking about, it is in Central America. Then he did his topic was his tissue culture, plant regeneration of cassava and tropical legumes. Second postdoc, he did it from Max Planck Institute of at Cologne, Germany. He was working on transcriptional regulation of nodulin genes in legumes. Right now, he is working in Biological Research Center, which is considered to be the center of European Center of Excellence. He is heading the group of Arabsis molecular genetics, and his citation is more than 2,000. Right, his edge index is around 2,000 something like that. So if you just Google it out and put it Laszlo Severos, you will come to know what great personality we are having out here. On the top of it, even he's the best researcher, nothing, no doubt about it. But over and above, what we require, the human heart, the human nature, the very human thing. So that human is here in front of us. So I thank Laszlo that you had take out your time to be here with us. So over to you, Laszlo. Thank you very much. We'll start cameras.
So in the next 50 minutes, I will talk about such aspects of our work, what we are doing in the last few years in Hungary. But first of all, I would like to make a short introduction where I am from. And just describe shortly this institute. As Nitin told it, this institute was established around 50 years ago, and there was an anniversary right now. And this is in Hungary, and in Central Europe. That time it was the best place to work. So when I started my PhD, I was happy to get there. This is an area image of the institute with the main labs, administration and some services. And the excellence of this institute is shown that the Nobel Prize of this year is Katalin Karikó, who he started her career in this institute. She was a student in Brooklyn, in local university, which is quite good in Hungary, and she started her career. So as our director, there was a type of celebration this autumn for Katalin, and this is the opportunity. So this is really not a bad place that time, and this is changing right now. And there is a new form by the government that they established, but still I think one of the best places to work biology. So what I'm going to talk about is the consequences of environmental stresses in plants and what we are doing. So if you know probably that there are some problems in the world and then number of such environments exist which is not really good for the plant. So you can face drought or cold weather, hot weather, soil salinity, sometimes inundations. And the most common thing that these extreme conditions are coming together. So very typical in an arid environment that there is high temperatures, where extreme temperatures there, and there is lack of water, and it's often combined with the soil salinity or toxicity. So plants has to cope with that on those conditions. Okay, so this is just a video what I find showing how climate is changing in the last 120 years. And yellow and reddish spots, they show the changes, not the actual temperatures, but how the changes is accelerating. And then you can see that when we are approaching today, it's more and more red and the globe. So that means that this process will be, I mean, the extreme environmental conditions will be probably more serious in the future. This tendency is going. So, and last year, we had also a serious drought in Hungary. So the field, the maize field in August, looked like this one. And so they lost 80% of their crop unless they could water for some way. So what we are working on, not maize, not crops, not rice, but mostly we work with Arabidopsis thaliana, a model plant in plant sciences. And if we reproduce all these conditions, soil salinity or drought, then the plants start suffering, and we can study all these physiological and molecular conditions and consequences in this plant. So if drought and salinity compare, there is a common component, and that is osmotic stress. And the secondary component is oxidative stress, which is actually the consequence of all extreme conditions and abiotic stresses. And salinity, it is still ion toxicity is something which is serious for plants. And the consequences are that the plants stop growing, cell division is declined, photosynthesis is effective, so this is one of the first which is shut down, and evaporation is reduced, and the accumulation of reactive oxygens, which is creating oxidative damage, is a quite characteristic of all these type of stresses and leading to cell death. But the plants can defend for a certain degree, and try to manage this condition. So if there is a lack of water, the stomata closure is one of main defense mechanism in plants. That the plants close stomata and try to evaporate less, but that affects photosynthesis also. So there are large-scale gene activation. The number of genes are induced or reduce their activity, and then you can find a number of metabolic consequences, like trying to make osmotic adjustments and scavenging reactive oxidative species. So the plants can cope with these problems to a certain degree, but there are big differences in between plants. So firstly, I try to talk about an aspect of our work, what we are doing for 2 decades already, and it is related to proline accumulation. Proline is an essential amino acid, but it has a number of other functions, and it is known for quite a long time. This is the first publication that they described that in a grass during wilting, amino acid are changing, and the proline is the one which is accumulating most. So it is from 58 to 54. So you can see that all these observations were described first quite a long time ago. And to see what is happening in the cells and what proline can do, and just want to revise shortly. So if we, in Arabidopsis model, we are making osmotic stress, then the proline accumulation takes place for a few days, and if we, for example, add water for these plants, then proline is concentration decline very, very fast. So how this is doing? Proline, the principal biosynthetic pathway is from glutamic acid, glutamate, and in 2 steps, proline is synthesized mainly in the cytosol. In some conditions, it can be a chloroplast. And the catabolism of proline takes place in mitochondria, again in 2 steps. So during stress, mainly osmotic stress, so drought, biosynthesis is upregulated. And the first and most important enzyme is so-called pyruvate 5-carboxylate synthase, or P5CS. I will talk about this. And this is which controls how much proline is synthesized. And the catabolism is again in 2 steps in the mitochondria. The first step is catabolized by proline dehydrogenase, or PDH. I'm going to mention this. So the biosynthetic pathway, which is principally a phosphorylation and 2 reduction steps, is induced during osmotic stress, and the catabolism is blocked by stress. But after stress, the catabolism pathway is activated. So this part is activated after stress release or recovery. So to study what is the importance of can be the importance of proline in plant's life, there were 2 approaches. One is to overexpress and try to accumulate proline in an excess way in plants. There are some experiments describing another approach. In Arabidopsis, we used mutants which disrupt biosynthetic or catabolic pathways. So if you see the P5CS mutant, in other words, there are 2 genes, but this gene, if it is disrupted by a mutation, that is, and we compare salt and salt stress to proline accumulation, you can see that there is no such accumulation or very minimal. But the proline dehydrogenase mutants are accumulating the same amount of proline as wild type or similar. And these mutants, in the lack of proline, they are hypersensitive to salt. They have a higher amount of peroxide accumulation. You can see in the DAP staining compared to the wild type, and higher lipid peroxidation. So it indicates that with salt, there is a higher amount of MDA accumulation that is more oxidative damage in these plants. So this we described years ago, and afterwards we were studying a number of aspects of this metabolic pathway, and we find that not only osmotic stress, but other stresses can regulate the proline accumulation. For example, high light. So if you put plants high light, that they accumulate proline not as much as osmotic stress, but several times higher than the control. And if we put the plants to darkness, and then we expose them to salt, then a proline accumulation doesn't take place. It means that this light has a positive effect on proline accumulation, and it is needed. So if we eliminate light, then the salt induction is not working. And what we find that with another group in our institute, that there are a number of factors which are already known and which mediate light signaling, and one of them is a basic type of transcription factor which is called Hi5. This is one of the central factors in the light regulation. There are several thousand genes controlled by Hi5.
Just screen share this. Screen share this.
Okay. So let's go back to high five. So this transcription factor was studied by another group in our institute, and they made a chip-seq analysis. So where this transcription factor binds in an Arabidopsis genome, and to our surprise, it was binding in the promoter region of P5CS gene, and also the polydehydrogenase gene. And in a separate experiment, we identified such mutative binding sites in the promoter of these two genes, which could be targets of this high five interaction. And with chromatinide immune precipitation, we confirmed that in fact the high five is binding to so-called C box or G box in the promoter of the P5CS gene, and also promoter of the protease gene. So that this shows that there is a direct link in the molecular level between light regulation and proline metabolism. And we checked the proline accumulation in the mutant. We find that the proline accumulation is lower in compared to the wild type, and also the P5CS expression is lower in the mutant than in the wild type plant. So that clearly indicates that this transcription factor is in fact important. Also, it is not essential for proline accumulation, but high level, yes. Okay, let's go a bit to the photosynthesis, because photosynthesis is one of is actually the main energy source of plants, and we find that proline is influenced proline metabolism by light. But is there, besides this light signal transcript signaling, is there some other aspects from the light, and or not? That was an interesting question. So this is just a scheme of electron transport, photosynthetic electron transport in the chloroplast. This is from textbook. Then you can see that there are complexes that PS photosynthesis, the two, then the cytochrome B, then the PS1 complex, which is carrying electrons gradually, and the not pH, reducing not pH is produced, which is used afterwards. Okay. So not pH is produced in the end, and then ATP is produced in another reaction. So this is just a textbook thing, and then this process can be blocked by several known inhibitors like DCMU, BBMB, and then paraquat, which has a bit different effect because it is taking electrons here from the FDL. So to imitate, to test whether if we block photosynthesis can somehow influence directly proline metabolism, we use the DCMU, which is blocking photosystem 2 in photosynthetic electron transport. And if we when we stimulated the plants with the salt, we could observe proline accumulation as usual, but in the presence of DCMU, this accumulation was only 20-25% compared to untreated. So that means that if we shut off photosynthesis by a drug, then consequence is more or less similar as we put it in the darkness. And in a molecular level, DCMU could induce the expression of proline dehydrogenase. This is a logarithmic scale, so this is much higher, the differences. And DCMU alone, so that means that shutting down the photosynthesis means the proline catabolism is enhanced. And photosynthesis was needed for P5CS high-level expression. Okay, this doesn't seem to be big, but this is a level scale, so it is a highly significant difference. So P5CS activation was much less, which was somehow similar to the darkness, and metabolism was also enhanced with this. So that this clearly indicates that there is a strong relationship between photosynthesis and proline metabolism, and this typically stress-induced pathway is rely on light signals and rely on also somehow signals coming from a photosynthetic electron transport. But what is the situation if we want to see how proline metabolism influence photosynthesis or not? So the reverse. So we used our mutants, P5CS mutants and protease, the catabolic mutants, and we tested a number of physiological parameters like photosynthetic electron transport rate or non-photochemical quenching, NPQ. And what was our surprise that, for example, photosynthetic electron transport rate was lower in both types of mutants. So this is a Y type, and this is the mutant. This is a significant difference. And when we put these plants to soil stress, the difference was even bigger. And the NPQ, which is also quite a typical characteristic, was in each mutant was higher than in a wild type. So that means that not the proline as a metabolite is important, or at least it is not. It is happening also, but proline, besides proline, the intact turnover, biosynthesis and catabolic pathway, that it is functioning, that is something very important. And if it is blocked, then photosynthesis is affected, which is probably the situation. This is just summarizing that proline is synthesized in through steps, and it needs reducing power. So it's reducing the net pH in 2 steps, 2 reactions, and photosynthesis is producing that. So when this process is blocked, then photosynthesis can be affected, but because in stress conditions, this net pH is not consumed, so the photosynthetic electron transport can, electron transport can be over-reduced, and this is producing then damaging reactive oxygens. And on the other way, excess proline can be consumed by catabolic pathway in the mitochondria, and then they can give electrons to the mitochondrial electron transport. So during stress recovery, this is a very important pathway giving electrons for stress recovery and giving this balance. So it seems that this turnover itself is also very important, not just the concentration of proline. And then light can directly influence expression of the protease gene. And it turned out that low energy condition, which is in the darkness or when we block photosynthesis, then it can enhance activity of the protease gene. There are some other aspects, but I don't want to complicate things more. So I was mentioning mitochondrial functions, which is also important for energy supply in plants and also, of course, in animals. And electron transport in mitochondria is the central core aspect of energy production. So there are, again, 4 complexes: complex 1, complex 2, then complex 3 and 4. And the electron is transported to all these complexes, and this is the so-called oxidative phosphorylation pathway, because finally ATP is produced by the ATP producing ATP synthase, which is embedded in a mitochondrial membrane. So it was already shown that the mitochondrial electron transport is a quite sensitive process during stress, and but there was not much information what is happening and why it is important. So with mutagenesis, programs, we identified 2 components of this electron transport rate. One is a protein which is called EPR40, which is associated to complex 3. It's not part of the core complex, but it's associated with that. And the so-called NOSF 8, which is part of complex 1. And we identified mutants. This is not a very new figure because this mutant we published years ago. The PPR mutant is a dwarf. And what was interesting that it was hypersensitive to abscisic acid. So it's a bit smaller than a white type plant. But when we had ABA, it is blocking much more the growth of this mutant and also germination. And one aspect that ABA is abscisic acid is promoting stomata closure. So if we had ABA, the white type stomata, they close, and the mutant, the stomata closure was faster. So it was this hypersensitivity to abscisic acid was not only in the growth or germination stages, but also in stomata closure. So you can see that the stomata pores, they close much faster in the mutant than in the white type. So we are thinking that if the stomatas are closing faster, that could be beneficial for drought tolerance, because this is one of the first aspects of saving water in the plants. So we compared, this is a relatively new result in our lab. We compared the mutant we had two alleles, one on knockout, another is knockdown, overexpressing plants and white type plants, how they survive water deprivation. And it turned out that the mutant survived much higher frequency than the white type or the overexpressing plants. So this is just a quantification of such results. You can see that in some conditions where the white type plants survived around 10%, this mutant survived around 60–70%. It's a much higher frequency. So that can, and this could be confirmed by. Okay, so okay, so let's go back. Then we characterized the effect of these mutants in a physiological levels and with plant phenotyping. And for example, when we compared photosynthetic electron transport rates, changes in the mutants and the white type during this water deprivation, this rate was dropping much faster in the white type plants than in the mutants. And also when we followed, I will explain a bit phenotyping, how this ES2 maximum quantum yield is changing. It was dropping faster in the white type than the mutants. So that means that the photosynthetic capacity of the mutants remains protected and more stable during water limiting environment. And we characterize a number of other physiological parameters. I just want to show lipid peroxidation rate in this moment. We can see the white type is the black. It is going up during water deprivation. It's a normal reaction. And in the mutant, the PPR1 mutant, especially this oxidative damage is much lower, around half than white type. So that means that this plant is somehow protected. It has higher relative water content after the water deprivation. So that could be the reason why these consequences are not as strong as in the white type plants. We made some metabolomic studies, just a few metabolites which are influenced by water deprivation. For example, you can see the proline accumulation in the mutant is much lower than in the white type plants. glutamic acid is contrary, which is dropping during drought treated plants. It is less affected in mutant, and glucose 6-phosphate, which is a central metabolite in sugar metabolism is higher in the mutant. So there are profound metabolic changes also which takes place. The other mutation, what other gene that I mentioned, this is a so-called, and they use FS protein. This is a core protein of complex 1 in mitochondrial electron transport. So then we identify the mutant, knockout mutant, and which grows more or less in the same rate as wild type plants, which is a bit surprising because the electron transport in the mitochondrial functions are reduced. It is not a knockout. I mean, it is a knockout mutant, but it is not eliminated completely. But when we compared these plants after water deprivation, we could find that these plants are less effective, less wilted than the wild type. So then we measured here again a few parameters like photosynthetic parameters, which these ones that we use routinely during these drought experiments, and we could see that this quantum yield, PS2 quantum yield in these plants are less affected than the wild type during osmotic stress or oxidative stress, not only in drought. And during drought, the electron transport rate is more or less the same in the wild type and mutant plants, in well water plants, but in drought conditions, the wild type electron transport rate was dropping faster than in the mutant. Clear indication that this mutation can help to stabilize photosynthetic processes. And there was less oxidative damage, which was osmotic stress or oxidative stress during oxidative stress, and the rate of plant survival was much higher again in the mutant than in the wild type plants after rewatering the plants after drought treatment. So you can see that it's more or less double or 3 times than the wild type. So it seems that both mitochondrial proteins, both of them are encoded in the nucleus, not in the mitochondria. So both of them are somehow important to maintain mitochondrial electron transport rate, mitochondrial electron transport, and which can block accumulation of reactive oxygens such as peroxide, and that makes plants in the mutant, when there is more peroxide in the mitochondria, we measure that that can promote stomata closure. So it is ABA is promoting stomata closure, but local peroxide content is something essential for that. It's a secondary signal. So when there is more peroxide in the mitochondria, this is not a damaging concentration, then the stomata are closing faster, relative water content is higher, so the plants are less damaged by osmotic stress, which implicates that photosynthetic functions are still better than in the wild type plants. There is less reactive oxygens in the whole plant and less oxidative damage, and then the survival is higher. Okay, let's jump to another field. Years ago, we developed a new genetic system where we used cDNA libraries from halophytic plant. So the Rapidium crassifolium, this is a close relative of Arabidopsis, and this can survive in a very saline soils. This is in Hungary. There are some saline soils, and in very extreme conditions, you can see this is the salt is precipitated. It's sodium chloride, sodium sulfate in the surface of the soil. So it's no other plants survive that except Salicornium. and this Lepidium. So we made a cDNA library from this plant in a plant expression vector controlled by estradiol inducible promoter. We wanted to have an inducible, well-controlled promoter. And we generated a number of Arabidopsis transgenic lines, around 50,000 lines. And we screened these plants for tolerance to salt, oxidative or osmotic stress. And we identified several genes which, when they were overexpressed by this from this plant, they could improve tolerance to some of these stresses. So this was the idea, and I'm going to show you one example which clearly shows the power of such a genetic approach. We find in one line that if we compare the growth, this is in vitro, this line and the wild type plants, they were very similar. Estradiol didn't really affect the growth. When we added paraquat to the medium, so oxidative stress, without estradiol, these plants were affected in the same way. But when we added estradiol, so we induced the inserted cDNA, this line, these plants could grow much better than the wild type. Really indicates that the induction of this cDNA could be beneficial and could alleviate the oxidative stress. And when we sprayed these plants in the greenhouse, they could survive while the wild type not. So that was very clear that the oxidative damage of this inserted cDNA would reduce oxidative stress. There are two lines, what we produce in QC, and the oxidative stress MDA accumulation is much lower than the wild type after the spray. So what we find that the inserted cDNA, it was very short, encoded a polypeptide or a small protein with only 69 amino acids, so it's very short, and only a signal peptide could be identified, nothing else in this. And what this small protein can do that the plants survive such harsh conditions? So we don't know yet the exact answer, but we try to localize with GFP fusions where this protein is localized in the cells, and we find that it is associated with tonoplast in the cells and probably the parasmatic reticulum. Now we are trying to do more precisely the association. We call this small paraquat tolerant protein. That's why this SPQ name. And we try to identify interacting partners with immune precipitation and mass spec identification. We identify some of them, which now we are trying to verify whether they are really implicated in these responses. And our other surprise was that besides improving tolerance to paraquat, this herbicide, the plants showed hypersensitivity to ABA. We tested the response to a number of hormones, and it turned out that they were more sensitive to ABA. So here you can see the stomatas, they closed faster in the overexpressing plants than in the wild type. And when we quantified this stomata closure, the stomatas were more closed after ABA treatment. And some of the stress-induced genes were expressed in a higher level than in a wild type. So it clearly indicates that ABA hypersensitivity could be observed in a stomata level, could be observed in a growth level, and could be observed also in a molecular level. So we tested a number of physiological parameters during water deprivation. So, for example, lipid peroxidation rates were lower, photosynthetic electron transport rate in well-water plants were very similar in the overexpressing plants in the wild type, but in the Y-type plants, this electron transport rate was dropping faster than in overexpressing plants. And we tested how these plants can survive water deprivation when plants were withdrawn by water for a certain period. Then they look like this, they're quite yield. And after rewatering, much higher frequency of this overexpressing plants could survive than the Y-type. This is just the quantification of two experiments. Once we use the mild or moderate desiccation, and other is a more severe. But you can see that there are 4 independent transgenic lines. They were surviving much higher frequency in both cases than the Y-type. So that indicates that it is important this overexpressing of this small protein could really positively influence growth responses. And in the last few years, we started to use plant phenotyping using image-based tools. This is just a summary what can be done. The advantage of this approach that physiological parameters are scored or not by destroying the plant, but following by different images. One is visual light, so it's color photography. Then it can be chlorophyll fluorescence measurements, can be hyperspectral images, which give some metabolic indices, and infrared imaging, which can give you information about the changes of the temperature of the plant without damaging the plant. So we have such equipment in our institute, and we can parallel and automatically measure growth and the physiological parameters of, in this moment, 480 plants. We can program this plant and program this equipment and then check the growth or different physiological parameters. So it's one typical experiment of growth experiment in this equipment is like that. We measure the soil water content, for example. When we water the plants, is more or less similar, or it's equal. When we stop watering, the soil content is dropping down. And around 20–25% in our condition, in our soil, that is more or less the limit of viability of Arabidopsis. So in this case, the plants start dying. And if we see how plants are growing, this is based on color images, then color segmented plants. So it is calculating green area actually, calculating rosette sizes. So the rosette sizes are growing up in well-watered plants. And if we stop watering for a few days, nothing happens, but then growth stops. And then this green area is declining when plants start dying. So this is more or less typical conditions. And when we compare these transgenic plants and Y-type plants, you can see these color segmented images that how are the rosettes. They are growing in well-watered conditions. Always the lower line is the Y-type here, different time points. And when we stop watering for a while, it is just a decline of growth what we can see. But then plants, when they are starting dying, then we can see that there is some difference between the Y-type and the transgenic plants. When we rewater the plants, higher frequency, high percentage of these plants survive than the Y-type. So this is just quantification of growth. You can see that the rosette size is dropping faster in the Y-type plants, and this transgenic plants, it is slightly lower. And when we see how the shape of the rosettes, which is a bit even more informative, you can see that the decline in the Y-type plants is faster than these transgenic plants. And when we see photosynthetic parameters, for example, change of electron transport rate, this is in time. So it's the last 5 days, what I show in the curve first. Electron transport rate is declining much faster in Colombia than in transgenic lines, and NPQ is enhanced faster in and the white type then in transgenic plants, which is indicating also that they are, the photosynthetic activity is more healthy. So we don't know what is the precise molecular function of this plant, this protein yet. We are working on it, but what is clear that it can block reactive oxygen accumulation and damage in paraquat treated plants. It is clear that it can enhance ABA sensitivity and promote stomata closure, promote expression of protective genes like dehydrins, and then reduce oxidative damage and enhanced protection can improve plant survival as was demonstrated by previous data. And now we are trying to overexpress these plants in this protein in some other plants. We used rapeseed, and the first results are promising. So this we used just a test in vitro germination test, and we could see that in presence of paraquat, for example, root growth or hypertrophic root growth of this transgenic rapeseed lines, it is higher than in the white type. And when we compared desiccation or the reaction of this, there are two transgenic plants in low water conditions in the white type, and after water depredation, these plants were visibly more affected than the transgenic plants, and their physiological parameters were also less effective. So proline accumulation was lower, probably due to higher water content, and oxidative damage was also lower in transgenic plants than in the wild type. So that is indication that this protein can work in other plants also. Okay, how much time I have still? Because we have a collaboration with in Bangalore with the NCBS Institute, and that is targeting epigenetic profiling of rapeseed. So it is a bit, when we started, it was also a bit new field for me. And so what we would like to see, what kind of epigenetic marks are changing in rapeseed during osmotic stress. We use polyethylene glycol for that. We tried also water deprivation, but the variation was too high. With polyethylene glycol, we could treat the plants in a much more uniform way. So after 2 days, they look like that. They don't like very much, but they don't die. So we were taking samples for 10 days and different time points. After this PEG treatment, we followed how is the proline accumulation. It is accumulating for a week, very, very high levels. So it was a type of physiological marker for us. And then we tried to see how histone modifications are changed in such conditions. So probably you know that the structure of the chromatin, whether it is closely tied or is more open, more prone for transcription, gene expression, it depends on histone modifications. Some histone acetylation is usually makes the chromatin more open. Methylation depends which residue can be favoring heterochromatin structure. So then we try to see how these changes are happening in the whole genome. So we were actually following methylation patterns in rapeseed histones and see that which type of methylation can influence. So we used H3K27 methylation patterns, which are repressive marks. So if this methylation is accumulating, the chromatin is more condensed, and H3K4 methylation that makes the chromatin a bit more loose, more prone for transcription. So we made a chromatin IP. First, we made transcript profiling after these treatments, what I showed you. After 6 hours and 24 hours of polyethylene glycol treatment, This just as the heat map of different repetitions, you can see that there are quite big changes. And a number of genes which were differentially regulated after these treatments were thousands. So we could find a number of induced genes and a number of repressed genes in both time points. It's just the Venn diagram how these genes were regulated. So this treatment was okay. We could find a number of stress-induced genes also. And then in the next, we made a ChIP-seq analysis. So we used antibodies which could recognize histone methylation in this K4 methylation or K27 methylation. This is opening the histone, opening the chromatin, and this can close the chromatin more. So it is just the distribution of this histone methylation, usually close to the transcription start site where there was this peak of this histone methylation changes. And then we tried to find in the genome, the whole genome, what kind of genes and which were methylated in this both sides. And this is just one of the P5CS genes in rapeseed. There are 10 genes, Arabidopsis only 2, but this is directing the proline biosynthesis. And what you can see that the mapping of this H3K4 methylation marks on this gene, that it is low methylation was characteristic in water plants and in the control plants, and this methylation was much highly induced, it's much higher during after PrEP treatment. Means that this gene is more prone for transcription during this osmotic stress. And then we compare these two patterns, the genome-wide patterns, how methylation patterns are changing and how the transcript levels are changing. And we identified a set of genes. I just show a few examples which were highly induced, and also the methylation was changed. For example, two P5CS genes, you can see that this yellow and red indicates expression after 6 hours and 24 hours, so they are highly induced. And this K4, H3K4 methylation was also induced, it was also higher. So in this case, and this 27, which is a repressive mark, was not effective. So this clearly induced that the overexpression or higher induction of this gene is associated with such epigenetic changes, which is favoring gene expression. And then there are several other genes like HO factor and dehydrins, transcription factors like ABF, which are known from Arabidopsis work that they are important for osmotic stress response, growth response. They were all upregulated in rapeseed also, and some of them were associated with changes in epigenetic level. And some other genes, for example, RBCS, which is associated with photosynthesis, this was repressed, and also, but epigenetic marks were also changing or not. So the correlation was not obvious, but in a few genes, nice correlation will be found, and now we are studying more detailed those genes. So in the end, I would like to show one aspect of the work, what gave us surprising results. Nitin was also involved in some of the experiments. So years ago, with this overexpressing genetic system, we identified a gene, it's Arabidopsis gene, a zinc finger protein, which when we overexpressed in Arabidopsis, that it made plants insensitive to abscisic acid. We wanted to find new regulators of ABA signaling. That was the reason of this work. So this is an estradiol inducible system. You can see that if we had ABA for germinate, it blocks germination in the Y-type plants and this line also. And then when we added estradiol to the system, this line, the plants could germinate by the Y-type node. So clearly, this can make plants insensitive to ABA. What was our observation? That the overexpressing plants were somehow smaller than the Y-type plants. So when we overexpress constitutively these plants in Arabidopsis. So this caught our attention, and we wanted to know why, whether it is related to ABA signals or not. And with this phenotyping system that I mentioned, we compared more precisely the growth of the Y-type plant and the mutant and some overexpressing lines. And you can see that the Y-type is growing more or less in a standard way, and the growth of these plants, which was we observed initially, was really lower than the Y-type plant. And we wanted to know why, what can be the reason behind this phenomenon. And we started to go to the cellular level. We made a scanning microscope images to see how is the structure of the leaf epidermis is changing, and which was our observation, and that the cells in these overexpressing plants were smaller and less differentiated than in the Y-type. This is a typical image in a Y-type epidermis. The cells are some of the cells quite big, and there are these lobes differentiated, and this plant in these cells, these plants not. So it could characterize with a special software. So the cell size was more or less half of this overexpressing plants compared to the Y-type, and the circularity of the cells was higher, so they were less differentiated, less complex. We made transcript profiling with this factor. I don't want to give the detail, but what was our surprise, that the number of genes which were involved in cell differentiation, cell wall biosynthesis, like expansin, extensins, and the number of genes which were implicated in root hair development. There's the RSR4, this is a transcription factor. It is well known that it is regulating root development, and other genes, they were downregulated in these overexpressing plants. So it seems that this transcription factor that we find in the ABA screen, that is repressing the expression of genes which are implicated in root hair development and cell wall development, cell wall biosynthesis. And some of with chromatin IP, we showed that some of genes are direct targets of this transcription factor also. Not all, but some of them, yes. So when we checked how is the finding the root hair formation of these plants, this transgenic plants has much less roots than the Y-type, and these roots were also shorter. So we could quantify the differences, and in fact they were shorter than in the Y-type, and there were less root hairs in the roots of these transgenic plants than in the Y-type. So we don't know yet completely what is behind it, but what we know that this is a model from a previous publication, a root hair initiation. There are a number of transcription factors and genes which are implicated. It's a quite complex field. It was also new for me, but there are two transcription factors which seems to have a very important regulatory role, the so-called RHD6 and RS4. And these transcription factors are regulating a number of other genes downstream. And what we find that in these overexpressing plants, especially the RSF4 was repressed. So, and some other genes which were shown by these people and that is controlled directly by this transcription factor. So probably this effect is that this transcription factor, this CFBA3, is repressing the expression of this other transcription factors somehow, we don't know. Well, it's binding also the protein promoter, and then a number of genes which are involved in cell wall biosynthesis and cell wall deposition and the root hair initiation. They are repressed. So this is more or less what I wanted to summarize our work and people who was involved and is involved or was involved in this work. I didn't talk with some aspect of the work, but this is more or less what we are trying to do and continue in the coming years. And we had a number of collaboration partners from our institute and also from abroad. And Nitin was involved in this ZFP morphological studies. And Professor Shiva Prasad from Bangalore is a collaboration partner with epigenetic studies in Ipswich. So thank you for your attention. If you have comments or questions, I try to respond.
Now I request all the participants. We are going to have a Q&A session now. If anyone is having any doubts, you can please come ahead and ask sir about their queries. Yes, you can just raise your hand and ask any query that you have.
So, what the duration of the lead leads to, how it will evaluate.
And it's still a consideration process. This consideration is and maintaining existing provide conditions and trans requirements because there is an enhanced and so this is the certified process which can be also very they show like high sensitivity response that nice and you see
That actually it has something to do with the polyethylene.
essentially you can try to see if it's there is some
Any more questions? Okay. Now I'll request Professor Dr. Laszlos. So I was saying is yes, any more questions?
Any other significant goal?
you, sir. Okay. I'll ask Laszlo sir to please have a seat on the dais. Now I request Principal Madam, Professor Dr. Zarin Patena, to come on the dais. Now I request Dr. Asmita Mistry, Head of the Department of Botany, to please present floral bouquet to Principal Madam. Now I'd like to thank Professor Dr. Laszlo Sevedos for giving us or enlightening us with the beautiful talk. Sir has explained how plants beautifully respond to abiotic stresses. Sir has enlightened us with different metabolic processes that the plant do, such as proline accumulation, mitochondrial respiration that the plants perform during stress. Sir has also helped us to identify different regulatory genes that play an important role, such as small protein transcription factors, and all these things were studied in Arabidopsis as a plant model. Thank you so much, sir, for such an informative session. Now I will request Principal Madam to present memento to Professor Dr. Laszlo Sevedos. Thank you so much, madam. Thank you, sir. I'll request Principal Madam to speak a few words about our college and our symposium.
Good morning, everybody. First and foremost, my apologies were here to the audience as well as were here to the guests for being late. I had previous appointments which I could not really miss, but I was definitely had made up my mind that I'm going to be there in this conference. And I am going to make sure that I'm going to hear what Sir has to say about it. Because the topic itself is so interesting that, you know, what happens in plants also happens over here to us, maybe on a different scale. Knowing how we respond to stress is something over here which we have to understand, because if you understand that it's happening in plants, it is happening in human beings, it's happening over here at molecular levels, at our and these lifespans also. So therefore, it's a very important topic. Second reason why it is so important was that in 1994, when my first project was going on, recognized by DST, Government of Maharashtra, not Government of India, Government of Maharashtra had given us the grant. I was working on the response of chitosan on plants, especially groundnuts. We had done all those work over here. It was more of an application type of a study. But one thing I wanted to know is that when I imbibed the chitosan molecule onto that of the seeds of groundnut, were the seeds having any type of a stress response? Because I was keeping it, and this was done over here in the rural areas. I think I had gone to Amravati, and there it was being done. So the reason was I wanted to find out that stress. At that time, I really did not know. I was not over here a plant biologist in any way. I'm a microbiologist. We work more with microbes over here and fermenters and organisms. So I said, how do I go about? So I started searching for papers. And one of the papers over here, I think it was this 1954 paper only over here, which says that if you want to detect stress in plants, you must find out over here the proline content. So we did the proline and the glycine content over here as such in terms of comparison with the control. A very simple experiment over here was done. We analyzed it, and where we found out that there were really not much of stress or changes which were happening. So this became a very important, and when sir was showing this paper, I was remembering the work that I had done over here in the labs that have now become your botany lab. And the same work, I remember I was so engrossed in that work that it turned out over here that it had become 8 in the night. I was alone working over here on that in the vacation, and everything was closed. And suddenly I started hearing noises. So I said, what the hell is this? I don't know what over here is coming up. I suddenly started thinking maybe over here there are some spirits coming out or what, because there were so many noises, and it turned out that these were those birds that were pecking onto it. So when I came out, it was pitch dark. Pitch dark. I couldn't see a thing because nobody had put on any lights. I was in that room alone, working in that lights. And the best part of it was I didn't know where the switches were. That was the best part. So I'm now searching for the switches. At that time, there were very little mobiles. I didn't have over here a light, and I'm banging into things over here and trying to find out where the switch is. Then I realized that there is a switch outside near our classroom. So I first put on that switch, and then I put on all the switches over here, and I came and I saw the time. It was 8:30 at the time over here in the night. So when you get engrossed in that of the work, I felt that this was the same passion that sir was showing into the way that work has been done. And it's really enthusiastically shown by him, which I'm sure all our botany students over here will have enjoyed and will emulate again and again over here when they are doing their work. A second thing that really made me very proud was the fact that there were 2 Indians over here as such who have worked with him. One of them over here is Nitin. And it's really encouraging that sir allows us to come to his lab again and again to work in the vacations and over here to learn the newer techniques over here as such. The digital imaging that he has shown over here as such is something which is nice, where we don't have to destruct the plant. Non-destructive means of analysis is something which is very important. And the use of the way that we can grow plants in stress conditions, in salty conditions, in salinity, where there is so much over here of things that are going on, especially when there is water shortage, which is going to happen and which is happening in India as well as in abroad. We will require this knowledge as to how over here to create those plant mutants which will help us to survive on that of this planet. So this is something which is really very interesting, and I request all my budding students in botany. Okay, botany is a small division of our college, but it is a department which is, you know, enthused to do work, which works over here and makes sure that the students over here do a lot of work for them. And I'm very happy over here to see this type of audience. I'm happy over here, and I'll be more happy if these students will go about taking more and more research. I'll be more and more happy if the students do not just do application research, but do fundamental research like the way sir has been doing, because just having application does not have much meaning until and unless you are able to prove with X, Y, Z right up to that of the cellular levels that this is what has been done. And this is the reason why over here it happens. So I request all of you all, especially because now it has become an NEP, the new education policy has come into place. There is internship. Tap him to go over here abroad, to see that over here the other labs over here are utilized. Go about over here finding out in India as well as in abroad where you can go to do your internship for the next 2 years. Okay, where in the vacations, select a topic that interests you. Talk with people over here as such who are working on that. India as well as abroad. There is no, most of us over here are most happy if some student comes over here and asks a question. Okay. We always over here answer them. and we answer them. I can assure you that, because I have gone through that and I know about it. You ask any scientist over here that this and this is happening, I don't know what is happening, can you pass me a paper on that? And I can assure you that in the next 1 or 2 days, you will get 10 papers similar to that over here attached to your mail. Because everybody knows that, the interest has to be cultivated in science, and young budding researchers require help, which they give in plenty. On this occasion, I would thank everybody over here who has come on this, worked over here so hard to make this a success, and best of luck to all of you. Thank you.
Thank you, madam. Now I'll announce the participants will have a break for 10 minutes for high tea, and we'll meet back again at 11:15 and start our second session. Okay, thank you.
Please be seated.
नाही यांच्याकडूनच नव्हते फक्त त्यांना काय करायचं स्लाइड शो होती नाही करायचं फक्त यांच्याकडून ती शेअर प्रेझेंटेशन बोलवते आणि कॅमेरा पाहिजे तर ऑन करू कॅमेरा त्यांना परत ठीक आहे एवढा ऐकला हे फक्त झालं की
हा लॉक करा नाही तर तो काय
Okay, now we'll officially begin with our second session. I would like to call Dr. Ravi Gupta on the dais. And now I'll request Dinesh Agre sir to come and introduce Dr. Ravi Gupta.
Good morning, dear all. Today, we have an eminent guest, Dr. Ravi Gupta with us. He is an alumni of Delhi University. He has done his graduation, master's, as well as PhD from Delhi University. Currently, he is working as an assistant professor in College of General Education, Kukmin University, South Korea. His research interest is in proteomic, growth regulators, and stress physiology. He has published more than 100 research papers in national and international journals with high impact factor. The cumulative impact factor is around 436. Total citations are 2,325. I-10 index is 57 and H index is 28. Sir has completed 2 research projects of grant around 1.20 lakhs and 1.48 lakhs US dollars. He is currently working on the project titled Establishing a Cysteine Redoxome Profiling Workflow to Decipher the Molecular Mechanism during rice magnoporthe or as an interaction. The project cost is around 1,44,000 US dollar. Sir is also working on editorial boards of various reputed journals like Springer Nature, Frontiers Media, Willy, etc. Sir has awarded by various scholarships like Ramalinga Swami Re-Entry Fellowship, Ramanujan Fellowship, Korea Research Fellowship, and many more. Sir has life member of International Plant Proteomic Organization. So such an eminent personality we have to deliver the lecture in this symposium. So we will definitely take the benefit of their lecture and it will be great for us. Thank you.
So thank you, Dr. Dinesh, for introducing me and other members of organizing committee, especially Dr. Nitin for having me here. And Professor Laszlo, he beautifully explained the effect of abiotic stresses on plants. And I'm majorly dealing with the biotic stress, like how plants respond if there is any pathogen or pest in surroundings. Okay. Okay, so to begin with, hello everyone, and annyeonghaseyo. So it's a Korean translation for hello. So the people who are following K-pop or K-drama or BTS, they must be familiar with this word. So yeah. So although I have been living in Korea for almost a decade, but I have learned only these few words like hello and thank you and so far, and somehow I'm managing to stay in Korea. So anyway, so okay, so today I'm going to talk about rice and Magna Forte pathosystem. So it's a kind of relationship. So I was from very beginning, from my bachelor's, I was so curious to know how two organisms belonging to two different kingdoms communicate with each other. Which kind of messages do they exchange when they come in contact with each other, and how the other partner decode those messages. Okay, so I was curious to know, like when Magna Forte or any kind of pathogen, when it comes in contact with their host, which kind of messages they exchange, and how the host plants decode those messages. Okay, so today's my talk is about those relationships that. So I'm basically focusing on rice and Magna Forte pathosystem. Okay. So coming to the first partner, that is rice. Actually, it doesn't need any introduction. We all know that it is a staple food that feeds more than half of the world's population, and it belongs to the grass family, the Poaceae, and it contains Oryza and Japonica cultivars. Okay, so in India we have Indica cultivar, and in Korea and Japan and China we majorly have Japonica cultivar. So japonica is basically more rich in starch. So it is more sticky, and so that's why, because of this parameter, it is more prone to infection. So starch actually attracts pathogens. Okay, so and uh, rice production is highly limited because of pathogens. So if we look at the common rice pathogens, okay, so if we look at the common rice pathogen, you will see that rice blast disease caused by Magna Forte oryzae, it can cause up to 100% of yield loss in the optimum conditions. Okay, and in addition, there are some other pathogens. So this rice blast disease, every year it destroys rice that can feed 60 million people. So you can imagine, like, if somehow we can try to restrict this disease, we can leap a big step in securing zero hunger, which is one of the 17 UN sustainable goals. Okay, so we are focusing on this. So if you look at the typical blast disease symptoms, it includes necrotic, that is cell death spots all over the leaves, panicle, and because of this, the photosynthesis is reduced, overall metabolism is reduced, and that leads to the production of fewer rice grains. Okay. So plants have developed a two-layered immune system that can be understood by this zigzag model. So I assume, like I was told, that audience here will include the undergraduate students. So I will explain the basics of this so that you people can understand what is plant pathogen interaction and how plant immunity functions. So this model was proposed by Jones and Engel in year 2006. So as per this model, the plant immune responses are activated because of the identification of some conserved pathogen signatures, like bacteria. Almost all bacteria contain a flagella. Okay, so plants can what plants can do? Plants secretes proteases in the apoplastic space that proteases can break down the flagellin protein that is present in flagella. Okay, so that after breakdown, it produces a 22 amino acid peptide, which is known as flag 22. flag is derived from flagellin, and because of it contains 22 amino acids, so it's flag 22. Okay, so those that flag 22, and in case of fungi, like fungi's cell wall is made up of chitin. Okay, so when there is a fungus attack, what these proteases or the chitinases enzymes are present in the apoplast. So what they do, they can break down the chitin and they can produce chitin oligomers. Okay, so that chitin oligomers and that FLAG22, they basically functions as a PAM, pathogen associated molecular patterns. Okay, so these are the conserved molecular signatures that are available in almost all kind of pathogen types. Okay, so plant can detect these PAMs because of the presence of some receptor proteins in the plasma membrane. So those receptor proteins are known as pattern recognition receptors, PRRs. Okay, so PRRs of the plants that are localized in the plasma membrane region, they can recognize the PAMs, leading to the activation of first line of defense, which is known as pattern triggered immunity. Okay, so this is PTI. So PTI responses are they are generalized responses, and they are not very strong. So they leads to the they leads to the formation of some generalized barriers, like they can induce the closure of stomata, they can induce the formation of callose deposition over the cell wall. Okay, so their purpose is to restrict the pathogen entry inside the host cells. Okay, so as I told you that this PTI responses are not very strong. So majority of the virulent pathogens like those that can cause disease. So they can overcome this PTI by secreting some effector proteins directly inside the host cell, host cytoplasm. Okay, so those, because of the secretion of effector proteins, this process is known as effector triggered susceptibility. Okay, and this again decreases the plant responses, plant resistance. Okay, and it increases the susceptibility. So this ETS basically is because of the secretion of effector proteins directly in the host cytoplasm. Okay, so in response to ETS, what plants do? Plants secrete some receptor proteins, intracellular receptor proteins, which are often the products of well-known R genes. You must have heard about the R genes, okay, resistance genes. So these are the receptor proteins, intracellular receptor proteins are the products of R genes, and they can recognize these intracellular effectors to trigger a more robust resistance response, which is known as ETI, effector triggered immunity. Okay, and this ETI responses are more robust, more powerful, and it can lead to the hypersensitive response, that is the killing of pathogen infected cells to inhibit the spread of pathogens. In addition, ETI responses can also lead to systemic acquired resistance, that is the induction of immune responses in the distantly non-infected cells. Okay, so ETI is relatively more stronger, but ETI responses are pathogen specific because these are induced in response to specific effector proteins secreted by specific pathogens. Okay, so this is just the simplified version of this model. So we have some PRR proteins in the plasma membrane, and these are PAMs. PAMs are also known as MAMs, that is microbe associated molecular patterns, or DAMs, because damage associated molecular patterns, because as I told you, like some molecular patterns like chitin oligosaccharides are derived because of damage of chitin cell wall. Okay, so that's why they are also known as DAMs. Okay, so these are basically recognizing the apoplastic space of the plants. Okay, so since this apoplast acts as the first battleground where the interaction between plants and pathogen takes place, or we can say it's the first site where the communication between plants and pathogens initiate. So we decided to go for the apoplastic proteome analysis. We were interested in identifying, okay, which kind of signals the pathogen secretes in the apoplast and which kind of signals the plant secretes in the apoplast. Okay, so what we did, okay, one more thing I want to emphasize that, okay, if we look at the Magna Proteorhizae life cycle, it also stays in the apoplastic region of the rice. Okay, initially, it's a Magna Proteorhizae is a hemibiotropic fungus. Okay, so initially it occupies the biotropic stage and then it goes to the necrotropic stage. Okay, so in the biotropic stage, it stays in the apoplastic space and it secretes various proteins in the apoplast. And later on, in the later stages of its infection, it invades the cells. Okay, so this gives us one more reason of doing apoplastic proteome analysis. Okay, so we decided, okay, let's go for apoplastic proteome analysis. For this, we first optimize the methods for the isolation of apoplastic proteins. So when we do some kind of subcellular proteome analysis, like if we are focusing on apoplast proteome analysis, or we are interested in identifying the nuclear associated proteins, so then we have to first optimize our protein extraction procedures in such a way that it should have a maximum yield and negligible contamination from cytoplasm or other organelles. Like if you are extracting proteins from apoplast, it should not have any protein from the cytoplasm or from any other organelle. If we are detecting any contamination, that means your isolation procedure is not optimized. It's not good. Okay, so what we did, we optimized the methods. We observed that the buffer condition containing 200 millimolar calcium chloride and 5 millimolar sodium acetate, it yields the best results in terms of yield. And for checking the contamination, we used glucose 6-phosphate dehydrogenase enzymatic activity analysis. So this glucose 6-phosphate dehydrogenase is a cytoplasmic enzyme, and its activity should not be ideally detected in the apoplastic region. So what we observed, that only 3% of or maybe less than 3% of the J6 PDX activity was detected in the apoplastic extract. So we thought, okay, this is kind of very negligible and it's acceptable. So we can use this optimized protocol for the high throughput proteome analysis. In addition, we also use some intracellular marker proteins and extracellular marker proteins to check for the contamination. So intracellular proteins were not detected in the apoplastic space extract, and extracellular proteins were enriched. So this again shows that the methods are working fine. So once we confirm that, okay, our methods are working fine and the yield is good, so we went for high throughput proteome analysis. So this story that I'm going to tell, it was done back in 2013, almost a decade ago. So at that time, only two-dimensional gel electrophoresis was the most accepted proteome analysis tool. And in addition, there was another gel-free tool that was emerging out, which was known as MUDPIT, that is multi-dimensional protein identification technology. Okay, so what we did, we used a combination of both gel-based and gel-free proteomics technology to identify both rice and magna-proteorhiza proteins. And in total, we identified 732 proteins. So I think 30% of those proteins were from the pathogen side, from the magna-proteorhiza, and 70% from the rice. And then when we checked their functional annotation, we observed that amorhiza proteins included various glycosyl hydrolase proteins and various proteases, esterases, while rice proteins were majorly associated with the plant defense. Okay, so this is heat maps showing the expression profile. So you can say that glycosyl hydrolases are secreted by fungus as well as plant. So this shows that glycosyl hydrolases plays key role in determining the plant pathogen interaction. In addition to these common proteins, we also identified one protein that was identified as uncharacterized protein. Okay, and its function was not known. So we thought, okay, these glycosyl hydrolases are well known, and then these defense proteins are also we know about their functions. So why not to focus on this uncharacterized protein? Okay, so we decided, okay, let's go and check what this uncharacterized protein is doing in this magna-proteorhiza. Okay, so we termed this uncharacterized protein as MSP1, that is magna-proteorhiza, not prot protein 1, or we also called it as magna-proteorhiza secreted protein 1. Okay, so this is how we discovered MSP1 in our plant pathogen interaction in rice. And what MSP1 is, MSP1 basically belongs to a ceratoplatinine family member, and it was these proteins were first discovered in ceratocystis. So that's why they are known as ceratoplatinine family proteins. And these are considered as phytotoxins. So they produces some kind of toxic effects on the host. So that's why they are known as phytotoxins. But what was the molecular mechanism of this phytotoxicity was unknown. And the members of these family are of low molecular weight. So as the MSP1, which is of 14 kilodalton. And as I told you, their functions were unknown. So we decided to further work on this protein. Okay, so to begin with, what we first checked, we first confirmed the secretion of MSP1 protein during incompatible and compatible interaction. Incompatible interaction means when there is no disease, okay, and compatible interaction means when there is a disease. So compatible interactions are from the virulent pathogens, and incompatible interactions are from the avirulent pathogens. Okay, so we did Western blots, and we observed that this MSP1 protein is only secreted during the compatible strain, that is during when there is a virulency. Okay, so we thought, okay, there should be it should be somehow associated with the fungus virulence. Okay, and then we went to probing the. We wanted to check. This Magna Proteorhiza really secrete MSP1 in the infected leaves. So for this, what we did, we infected rice leaves with Magna Proteorhiza and probed them on the PVDF membrane, and we used anti-MSP1 antibody. And then here you can see that this fungus infected leaves show secretion of MSP1. Okay, so these two simple experiments confirm that okay, MSP1 is secreted during compatible interactions when there is a disease, and it is only secreted when it comes in the contact with a plant. Okay, and we also did some in vitro experiments, and we could not find the secretion of MSP1 during the plate culture of Magna Proteorhizae. Okay, so first we do like it's common for all the fungi, like you have to do some culture for fungus. So you can also check the secretion of MSP1 or any kind of protein during the culture stage itself. So we observed that MSP1 was not secreted during the culture stage. It was only secreted when it comes in the contact with the its host. Okay, so that was an indicator that okay, MSP1 has to do something with the pathogen virulence. Okay, and we also checked the secretion of MSP1 in the apoplast and cytoplasm. So we wanted to know that whether MSP1 functions as a PAM or an effector. So if it is an effector, it should be detected in the host cytoplasm. Okay, and if it is a PAM, it should only be detected in the apoplastic region. So we did Western blots. We isolated apoplastic proteins and cytoplasmic proteins from the rice after infection, and then we checked the MSP1 by anti-MSP1 antibody, and we detected that MSP1 is only present in the extracellular region, in the apoplastic region only. That shows that MSP1 is potentially functioning as a PAM. It's not functioning as a effector. Okay, so these experiments shows that MSP1 is potentially associated with the fungal virulence, and it is potentially functioning as a PAM. Okay, so we decided to characterize this protein further by analyzing the PTI responses. So what we did, we checked the reactive oxygen species ROS accumulation during extracellular MSP1 treatment. We also checked the cell death in the rice suspension cell culture. So for this, we used OC cell. This is the very common rice cell line. It was established in 1946 by some Japanese scientists. Okay, so we used this rice suspension culture, and then we observed that if you treat these rice cells with the recombinant MSP1, okay, so for generation of recombinant MSP1, we cloned it, we cloned it in a bacteria, we purified a recombinant protein using some affinity tags, and then we treated these rice cells. So we observed that increasing time after MSP1 treatment leads to higher accumulation of reactive oxygen species. In addition, MSP1 treatment also induce cell death. Okay, so for cell death, we use trypan blue staining, and for ROS accumulation, we use DAB staining. Okay, so this is ROS marker, this is cell death marker. Okay, and in addition, this MSP1 treatment also leads to ion leakage. That means ion leakage is also a parameter to measure the cell death. Okay, that means the membranes are weakening down and then the cell content is coming out. Okay. So we observed from this experiment, we observed that extracellular MSP1 treatment leads to accumulation of ROS. So this ROS accumulation is a PTI responses. Whenever there is a pathogen attack, you can see ROS burst. That is, ROS burst means a sudden and transient increase in ROS concentration. Okay, so that is ROS burst. So we observed that ROS burst in response to MSP1 treatment. In addition, we also observed cell death in response to MSP1 in the rice leaves. So to check the cell death in response to MSP1 in the rice leaves, what we did, we used cell death marker gene, that is PBZ. That is a cell death marker gene in rice. So we used PBZ promoter, and we fused this promoter with the GFP. So GFP is a green fluorescent protein. Okay, and so GFP will only be visualized if there is a cell death. Okay, because we fused it with the cell death marker gene. Okay, so we observed that if we treat MSP1, we can see that these green fluorescent signals that shows there is a cell death. Okay, so we observed cell death in at very high concentration. So anyway, these experiments suggested that MSP1 treatment leads to ROS burst and cell death in rice. Okay, in addition, we also check the effects of various biochemical inhibitors like protein synthesis inhibitor cyclohexamide and protein phosphatase inhibitor okadaic acid. So we observed that if we treat cyclohexamide along with MSP1, there is no cell death. So that means protein synthesis is required for MSP1 induced signaling. In addition, we observed that okadaic acid treatment markedly enhanced the cell death. That means okadaic acid is an inhibitor of protein phosphatases. Protein phosphatase are involved in the protein dephosphorylation. Okay, so that means that protein phosphorylation events are required for MSP1 induced signaling. Okay, so based on these experiments, we proposed a model. As per this model, manopautorizer secretes MSP1 protein, and it is identified by unknown PRRs. Okay, we don't know what is the MSP1 receptor, and this MSP1 receptor binding leads to the ROS production and some unknown phosphorylation events. Okay, and we also don't know what are the downstream signaling components. Okay, so till this 2016, we were aware that MSP1 treatment leads to the ROS production. It induces PTI signaling in rice. It induces cell death in rice. Okay, so what are the molecular components and what are the potential MSP1 receptors were unknown. Okay, so we decided to do a high throughput proteome analysis in order to identify the MSP1 induced signaling components in rice. So what we did, we used MSP1 and we used FLAG22. This is the bacterial PAM I was talking about. So we use FLAG22 as a positive control and MSP1. So this we know that this is a positive control. This is a well-known PAM. So that if MSP1 is a PAM, it should follow the same pattern of signaling as induced by FLAG22. Okay. So we observed that MSP1 treatment leads to the expression of PR proteins both at the transcription level. These are qRT-PCR and both and at the protein level. So these are Western blots. In addition, MSP1 treatment leads to ROS burst. So this experiment confirmed that our MSP1 treatment conditions are working fine. And once we are confirmed about these treatment conditions, we did the high throughput proteome analysis using a label-free quantitative proteomics approach. Okay. So this we did some multivariate analysis, and for this we use a MaxQuant and Perseus platform that was developed by Max Planck's Institute of Biochemistry, and I had an opportunity to learn this platform from the developers. Okay, so what we did, we generated some multi-scatter plots which showed that a Pearson correlation coefficient of more than 0.9. That means high reproducibility of the obtained data. In addition, we did some clustering analysis, and then this is the MapMan analysis. So MapMan basically tells us about the functions of the identified proteins. So we observed that proteins associated with the auxin and jasmonic acid and these peroxidases, loss detoxification are increased. So that means increase, green means decrease. And then proteins associated with the cell wall are majorly decreased in response to MSP1 treatment. We also did interactome analysis, and this interactome analysis identified the key roles of glycosyl hydrolase and peptidases. But from this experiment, we get a hint which kind of signaling is activated in rice in response to MSP1 treatment. But through this study, we did not find any MSP1 receptor. Okay, and we were more keen in identifying the receptor in particular, because if we know the receptor, we automatically know the downstream signaling components. Okay, so we thought, okay, there should be some loopholes in this study, so we decided to do proteomics again, but this time with the plasma membrane enriched fractions. So because PRRs are localized in the plasma membrane, so we thought, okay, those proteins are maybe low molecular weight, and they may be low abundance. So if we do the whole cell proteome analysis, we could not detect those. Okay, so what we did a plasma membrane proteome analysis. So for this, we enriched the plasma membrane proteins. We isolated the plasma membrane proteins using an ultracentrifugation method. And this time we use a TMT-based proteomics approach. So this TMT-based proteomics approach is more sensitive and robust than the previously used label-free approach. But the problem with this approach is multiplexing. So this is actually a benefit also and a problem also. So in what we do, we isolate the proteins from different samples and then we pull them together. And then peptides from different samples are labeled with some isobaric mass tags. Okay. So because of this multiplexing, normalization of data is very crucial to get a meaningful result. So we decided to do a two-step normalization procedure. One within the technical replicates of the sample and other between the biological replicates. Okay. And we checked the quality of isolated plasma membrane proteins using some marker proteins. So plasma membrane proteins were enriched, while this mitochondrial marker proteins and cytoplasmic marker proteins were not detected. So this shows, okay, our methods are working fine. So using this proteome analysis, we could identify some proteins associated with signaling, and those proteins were we already know about the previous label-free analysis. In addition, we identified 48 receptors or receptor-like kinases from this plasma membrane proteome analysis. But out of those 48 receptor-like proteins, only 3, that includes a chitin elicitor binding protein, a chitin elicitor induced receptor kinase, and a CDPK. So except for these 3 proteins, all other proteins were not changed significantly in response to MSP1 treatments. Okay. So we thought, okay, there again, we are missing something. So, and then when we searched more, we realized that receptor proteins are mainly activated because of their phosphorylation. And the same thing we observed during our okadic acid treatment experiment also. So we thought, okay, there is some role of phosphorylation in the MSP1 binding. So what we did a phosphoproteome analysis, and we isolated the plasma membrane proteins, and then we enriched the phosphopeptides using titanium oxide enrichment kit. And from this, we could identify 1,906 something phosphopeptides. And we did multivariate analysis. And of this multivariate analysis, this cluster 4 and 5 and 6 were of particular importance because this shows MSP1 induced phosphorylation of these particular proteins. Okay. So we, based on this phosphoproteome analysis, we generated a model. We updated our previous model that we generated. So in that model, you could remember that multiple things were unknown. But here we updated this model based on the plasma membrane proteome analysis and phosphoproteome analysis. So as per this model, MSP1 is identified yet unknown receptors. The receptors were still unknown by then, but we know that MSP1 increases the activity of RBOHB. This is a protein that is NADPH oxidase. So it is involved in ROS burst. So when there is a ROS burst, this protein is activated. So we observed MSP1 is activating this RBOHB protein, which leads to activation of CDPK, that is calcium dependent protein kinase. This calcium signaling is activated in response to increased calcium concentration. In parallel, we observed the activation of jasmonate biosynthesis pathway. So jasmonate and salicylate are two well-known stress hormones that participate in the plant pathogen infection or providing plant immunity. In addition, we also observed increased phosphorylation of receptor like cytoplasmic kinase 185 that leads to the phosphorylation of MAP kinase 1, which in turn leads to the phosphorylation of MAP kinase 4,5 and then MAP kinase 3,6. So because of the activation of this signaling, these signaling components, the gene expression is changed, which leads to the generation of PR proteins or some other metabolites involved in plant defense. So again, we were failed to identify the receptors, MSP1 binding receptors. So we thought, okay, what's now? I mean, where is the loophole? So we did label-free analysis, we did TMT-based proteome analysis, we did this with apoplastic proteins, we did with total proteins, we did with the plasma membrane isolated proteins. So, and then we thought, okay, there could be some problem in MSP1 perception by rice leaves. So rice leaves are so rugged. It's so difficult to treat something on rice leaves because it is covered with the cutin. Okay, there are lots of wax deposition over the rice leaves. So if you treat something, it may not go deeper inside the rice leaves. So we thought, okay, this could be one of the reasons. So what we did, we generated the MSP1 overexpression rice lines. So both that secretes MSP1 in the extracellular region and that secretes MSP1 in the cytoplasmic region. So we generated the constructs and then we checked their efficacy in the Nicotiana tobacco, and we observed that EMSP1, that is the MSP1 which is secreted extracellularly, which is apoplastic localized, can induce cell death and ROS accumulation in tobacco leaves. Okay, so once we are confirmed with the efficacy of our construct, we transform them in the rice. Okay, and we check the MSP1 expression in the transgenic lines by Western blot, and these are the confocal microscopy images that shows the expression of EMSP1 in the extracellular region. In the you can see green fluorescent signals in the outer cell surface only. Okay, and this is CMSP1, that is, you can see the GFP signal inside the cells also. Okay, so this experiment shows that, okay, our targeting is working fine, and this MSP1 expression we checked by qRT-PCR also. So from this experiment, we could identify 19 receptor-like kinases that were changed in response to MSP1 expression. So out of these 19 receptor-like proteins, any single or two proteins could be MSP1 receptors. So what we had to do now, we had to narrow down our target from these 19. So we thought, okay, this could be very tedious because doing co-IP or complementation assays or interaction studies with the 19 proteins would be so tedious. So what we did, we compared these 19 receptor-like proteins with the transcriptome data. Okay, in response that was generated in response to Magna-Porter treatment, and we could identify only a single receptor, that is MRH1, that was induced both in the transcriptome study in response to Magna-Porter treatment and in response to MSP1 treatment that we identified in our proteome study. Okay, so we thought, okay, let's focus on this MRH protein first, and then if it turns out to be a negative interactor of MSP1, then we can focus on other 18 proteins. So first we check the expression of this, reconfirm the expression of this MSP1, this MRH protein in response to magnapolymerase treatment. And we observed that, okay, the expression of MRH1 is increased after 72 hours post inoculation. So that means it has something to do with the magnapolymerase perception. So what we did, we then tried to check the physical interaction between MRH1 and MSP1. So whether these two proteins really interact with each other or not. So what we did, we generated the constructs. So we tagged MSP1 with the HA tag and MRH with the MIG tag, and we transformed these constructs in the rice protoplast. For this, we isolated protoplast from the rice cells, okay, and then transformed these constructs in the rice protoplast. And then we used anti-HA antibodies and anti-MIG antibodies, and we observed that physical interaction is available between MRH1 and MSP1. Okay, so this shows that, okay, these are really interacting with each other in the rice apoplast. Then we were interested in knowing which segment of MRH1. Okay, we sorry, this is this MRH we also termed as MBR, that is MSP1 binding receptor. This is the name that we give to MRH1. So which segment of MRH1 is interacting with MSP1? So for this, we check we did bioinformatic analysis of MRH1, and we observed that it contains a N-terminal signal sequence, a transmembrane domain, and a kinase domain. So we generated three truncated constructs, one with the kinase domain only, one with the kinase domain with some transmembrane domain, and one with the kinase and transmembrane and extracellular domain also. So from this co-IP analysis, we observed that MSP1 is interacting with the extracellular domain. Okay, and which is quite logical also, because MSP1 is secreted in the apoplastic space, so it should be interacting with the extracellular region of the MRH1. The kinase domain is usually available in the cytoplasm region, and it is involved in the activation of phosphorylation events in response to any binding, okay, signal binding. So through this experiment, we confirmed that MSP1 is interacting with the extracellular domain of MRH1. And then we reconfirmed the binding of MRH with MSP1 using BIFC. So BIFC is a technique that is in this technique, what we do, we take a fluorescent protein, either green fluorescent protein or yellow fluorescent protein. We split into two halves, and then one we tag with the one protein, our protein of interest first, and then other half we tag with the second protein. Okay, and if those proteins are interacting, the green fluorescent protein will be full and it will show the signal. Okay, so we observed that a positive signal in response to MRH and MSP1. So this shows that, okay, inside the rice leaves also these proteins are interacting. So first we confirm the interaction of MRH and MSP1 in the rice protoplast, and then we reconfirm that in the rice leaves. And then we were interested in knowing which part of MSP1 is basically active and it is involved in the MRH1 interaction. So we checked the we did bioinformatic analysis, and we observed that MSP1 contains total of 6 domains. Okay. So out of these 6 domains, what we did, we generated the synthetic peptides out of these 6 domains, and we treated those on the rice leaf. And this background was again PBZ and GFP tag rice lines. So if there is a cell death, we can see fluorescent. So we observed that, okay, domain 2 is inducing a high level of cell death. In addition, a slight cell death was also observed in domain 1. So we thought, okay, this is the domain 2 of MSP1 that is basically active, that is playing the major role. Okay. Then to reconfirm what we did, we used only domain 2 instead of the full length MSP1, and we treated the domain 2 on the rice suspension culture cell, and we observed accumulation of ROS in response to MSP1 treatment. Okay, and cell death in response to domain 2 treatment. So this confirmed that okay, domain 2 is the active domain of MSP1. Then we did some docking studies to know the key amino acids that are associated with the functions of domain 2. Okay, so we observed that okay, docking study further confirmed the interaction between MRH and MSP1 protein, and this is basically the domain 2. So domain 2 is associated with the interaction between MRH and MSP1. Okay, and we observed the role of 3 amino acids, D41, K49, and Q55. Okay, so we generated at least 10 models, and those 10 models suggested the key roles of these amino acids. But one model, that is model 6, it shows that Q55 doesn't have any role in the interaction. So we wanted to further cross-check the role of these key residues in MSP1 and MRH1 interaction. So what we did site-directed mutagenesis. We replaced D41, K41, and Q55 with the arginine residues, and then we rechecked the depth, the ROS accumulation, and we observed that okay, if we replace D41 with arginine, there is no difference in the ROS accumulation. If we replace Q55 with arginine, there is no difference in the arginine ROS accumulation. But if we replace lysine with arginine at 49th position, the ROS accumulating activity of the domain 2 is lost. So that means this lysine is kind of the key residue, okay, that is required, or that it is present in the active domain or active site of the domain 2 of MSP1 that is associated with these functions. And in addition, to further reconfirm the role of MRH in inducing the PTI signaling, okay, if MRH is the real MSP1 receptor, it should also be associated with the PTI responses. So we generated the MRH1 overexpression rice lines, and we observed that okay, MRH1 overexpression leads to the accumulation of reactive oxygen species and generation of secretion of PR proteins. Okay, so this further confirms that MRH has something to do with the plant pathogen infection. And then MRH1 overexpression lines also showed enhanced resistance to rice blast disease. Okay, so for this, what we did, we used the virulent MRHI strains. We inoculated with the wild type and overexpression lines, and we could observe a slight yet significant difference increase in the resistance against blast disease. So based on our whole study, we generated this model. So we are actually currently validating this model. So far we know that okay, MBR or MRH1 is the MSP1 interactor, and we know about the downstream signaling component. So, but we are trying to decipher which kind of phosphorylation events it initiates and which kind of MAP kinase signaling, calcium signaling, and hormone signaling are activated that leads to the activation of PTI. So this is we are currently validating this model. So that was all for my today's talk, and I would like to thank my team members. He is my postdoc professor, Professor Sunte Kim. He is in the Pusan National University, and we also have a collaboration with Professor Randeep Rakwal. He is from University of Tsukuba, and this guy, he is basically associated with the MRH data, and he is still doing his PhD on MRH1, and hopefully he can submit soon. And okay, recently we got approval from the government, Korean government, to grow LMO, that is living modified organisms. You must have heard about genetically modified organisms. Okay, so these are LMOs. So we can now cultivate LMOs inside our campus and the PMU. So that was back in 2021. So it was kind of recently. And this is the campus where this all work has been done, and our team members. Okay, so that's all.
Thank you so much, sir. Now I would ask participants if they have any questions. We are open for question and answer session. Yeah, go on. Mic on.
I think for biology, we don't have any similar mechanism. Like PJ is also activated in response to some of the capsid proteins of viruses, but those are not efficient. So we scientists are trying to look for some other mechanism, a different kind of signaling is activating in response to virus. Okay, and that is completely different from PJ and EPJ. So it's not, although it's not well understood, but yeah, you can say similar kind of signaling activated in response to capsid protein.
And sir, as MSP induces cells that which activate what? So can it be done by restricting the number of cells entering photosynthesis? Number of cells entering photosynthesis.
Number of cells of right?
Number of cells which enter photosynthesis.
Yeah, so yeah, it's somehow dealing with the subject that's correlation that's
Okay, any more questions? I would like to thank Dr. Ravi Gupta for enlightening us with the role of MSP1 protein, which plays an important role as an eliciting factor in rice and making us understand how the plants deal with different types of biotic stresses. Once again, thank you so much, sir. I would like to invite Dr. Asmita Mistry to present Momento to sir.
स्क्रीन लगाइए इंटरनेशनल सिंपोजियम
Screen. Thank you so much, sir. Thank you, madam. Okay. After two wonderful offline lectures, we are going to have our online session now. And for that, I would like to invite Tawfiq Sheikh from Department of Botany. He will introduce our next speaker, Dr. Riaz.
Thank you, Sharon ma'am. Good afternoon to all present. It gives me immense pleasure to introduce Dr. Riazuddin. Dr. Riazuddin has completed his undergraduate and postgraduate studies from Department of Botany, Delhi University. He obtained his doctoral degree from University of Zec, Hungary, Europe. His thesis for doctoral degree was overexpression of Arabidopsis glutathione peroxidase like 5G in salt tolerance. Speaking about his research recognition, Dr. Riazuddin has more than 18 papers present in highly impact factors international journals, more than 20 papers and posters in international conference, symposiums, and congresses. His citation score is more than 450 and H index score is 11. His cumulative impact factor is more than 90. His area of specialization is plant physiology and molecular biology. Sir have received many fundings and awards. One of the award which he recently got a seal of excellence for his postdoctoral work. Currently, Sir is enrolled for his second postdoctoral in University of Miguel Hernandez at Institute of Bioengineering. Now I request Dr. Riazuddin to please enlighten us with a wonderful talk. Thank you so much.
I will request I'll request Dr. Riazuddin to please start with the lecture.
Good morning to all of you, and a very early good morning from Spain. Now it's 7 o'clock in morning, and thank you so much for inviting me here. I would like to thanks to all organizing committee, and special thanks to Dr. Nitin Labane, who is having me here. So today, before me, there was two very fantastic talks delivered by Professor Laszlo Sabados and Dr. Ravi Gupta. They were talking about abiotic stress, and after that, Ravi Gupta told us about the biotic stress. So today, I'm also going to talk some of parts of my research, which I have done in during my PhD about the salt stress. So, as you know, like both abiotic and biotic stress both are very important to address in the present scenario because they have the negative impact on crop. Because of that, there are certain data available on the US Department of Agriculture and FAO, which stated that 50% crop reduction is happened because of biotic and abiotic stresses. So here I will talk only about the abiotic stress. In abiotic stress, there are many, but I will more focus on salt stress. So according to the FAO data, so there are 37% land around the global which are affected directly or indirectly by salt stress. And among them, 20% salt affected lands belongs to the fertile land, which is now getting saline, and it's fall under three categories like saline, sodic, and saline sodic. As you can see in the map worldwide, there are all over the world is a major problem, and it's very important to address this issue. So what is the salt stress? Salt stress is basically accumulation of the salt particles in the water or in the soil, which directly or indirectly impact on the plant health and induce the cell death. And it also cause one of the major cause to lose the crop yield. As you can see here, this is the graphical representation of the salt, and there are like few pictures, and I can say this is like white crusties all over field, and this is like very saline, and is affecting the soil health and impacting on the plant's productivity or overall health. So how salt stress impact on the plant's health? So basically it's induce there are many way to impact, but I will briefly explain how the salt stress uh, did in the plants. It's inhibit water uptake, it can inhibits the leaf growth, plant development, it can inhibit the photosynthesis, protein synthesis, leaf, it can induce the leaf senescence, and in the and overall because of this cumulative, this can also impact on yield performance. So one of the major impact of salt is like it induces the ROS, and accumulation of ROS leads to the oxidative stress, which can cause cell death. So another question is like when the plants comes in contact with the salt stress, what's happened inside the plants? So this is the diagram. This is like a model how plants. This is the one possible approach. Like when the plants comes to contact with salt, so it's the first response will be rose formation, and more rose formation, and rose accumulation will be there, which is like imbalance, and it can induce the oxidative damage, which can lead the cell death. But in response of that, plants has very sophisticated plant defense system, which is consist of two parts. One is like enzymatic. In enzymatic, there are I think everybody knows about this superoxide catalase and other antioxidant enzymes, and in another part is a non-enzymatic, so which can be mostly with like ascorbate, glutathione, and proline, polyamines, and etc. So in this while we were working on salt stress, so in nature like is nature is full with mysterious, and in nature there are few plants which are very salt tolerant. We said to halophytes, they are and another is glycophytes. Glycophytes are salt sensitive, and halophytes are salt resistant, which can complete their life cycles. So in these cases, I found one very interesting paper in 2000 from 2013. Bals and Dinkar, they stated that they did a comparative study at the level of gene and protein level, and they found like this Halonjala is a halophyte which was more tolerant, even 600 millimolar concentration, which is highly salt concentration. But if you see in the Arabidopsis, the plants already died. So what could be the reason, like why these halophytes are more salt resistant compared to the glycophytes. So they found that they in halophytes this antioxidant defense system working at the up regulation level, and also they found this sodium and potassium ion homeostasis was very well regulated in halophytes. And after that, they found very interesting one isoenzymes. This is called glutathione peroxidase. Is also a known is enzymatic antioxidant, which was mainly responsible for this salt tolerance in Thalangella. So based on that, we developed our hypothesis, and we decided to go with the GPXLs, which is known as glutathione peroxidases. And what is the glutathione peroxidase? They are non-heme-thiol peroxidases which contain cysteine in plants and selenocysteine in animals. They are important antioxidant enzymes. They utilize glutathione or thiodoxine as a reducing to reduction of hydrogen peroxide into the corresponding alcohol or water molecules. They have various VAS function. They have a role in detoxification. They have a role in signaling. They have a role in redox regulation. Only the difference between the animal and the plants is in their active site, only the selenocysteine in animal and cysteine in plant. So now, because I'm talking about the glutathione peroxidase isoenzymes 5 here. So why I choose only the 5? Because in the Arabidopsis there are like 8 isoenzymes which are located at several places. But I'm talking about this 4 because this 5 is very less characterized, and it was inserted in the inner membrane, plasma membrane. And also we found, or also based on the previous literature, in halophytes, GPXL5 is essential in the effective salt tolerance. That's why we focus on GPXL5. So in this scenario, we have several like aims, like what can be the question. So we propose like 3 main questions, like has this ATGPXL role in the regulation of ROS, redox homeostasis, and the maintenance of cells vitality, and what is the effect of decrease or increase GPXL5 expression on the activity of antioxidant mechanism in control condition and after the salt discharge as well. Have the GPXL5 overexpressing or this tDNA insertion ATGPXL5 mutants plants altered glutathione redox potential. And also we were very curious about to know like has this the ATGPXL5 any kind of function in the growth and development of Arabidopsis seedlings under control as well as after applying the salt stress. To follow or to achieve our aims, we we part our experiment in 2 parts, and we use only this Arabidopsis plants, and in Colombia wild type we also introduced this ATGPXL5 mutation lines and 2 ATGPXL5 overexpressing lines. So 2 experimental systems were utilized to address our aims. In the first experimental system, it was in plates. We used like half MS media, and we detected or measured the parameters for the ROS and the chlorophyll and anthocyanin content through the plant size software or image software, which was developed by Professor Laszlo Sabado's lab, and it is very, very good, I would say. And in another second experimental system, it was in this case is like 6-week-old plants we use and 100 millimolar NaCl for 24 hours. And we measure biochemical and physiological and enzymatic and non-enzymatic components. So in the first part, from the first experimental system, we investigated the role of GPXL5 isoenzyme in ROS homeostasis using the mutant seedlings. So the first, when we went through this microscopic image, and we found DHE fluorescence, it was, if you see, is compared to in the Colombia, if you see, it was higher. It's increasing when the concentration is increasing compared to the Colombia col 0 under control condition. But after also in the control condition, if you see, this ATGPXL5 seedlings has more superoxide radical, even in control condition. compared to the Columbia Coal Zero. And if you see after 100 millimolar NaCl, ATGPXL has highest superoxide radical content. So what does it mean? If you see here, you can see it's like in the control, and it's like highest is here. See, you can see the DHE fluorescence intensity. So these results, what they indicate? They indicate the role of ATGPXL might play an important role in superoxide radical homeostasis. After that, we check the total rows by using the another different dye, H2DCFDA, and in this DCF fluorescence we also measure, and we found that there were no differences in the root, but in the soot, if you see in the control condition, ATGPSL5 mutants soot has higher superoxide, so has more ROS level compared to others. As you can see in the images, also it was there were in root is like similarly we found it, but there is no significantly differences. But if you see here, it's like more, so we can say it has a role in fine tuning the post production. So there is a in theoretical there is a if higher ROS or superoxide there is there, so it's a impact on the cell's vitality. So that's why we check the cell vitality through the fluorescence marker, and we use the FDA fluorescence, and we found that in the root, the control condition, Columbia Coal Zero, after 100 millimolar concentration, it's going down. Vitality is going different. And if you see here in the mutant also is going down. And also, if you see in the soot, this is going also down. And after, if you see in the control condition compared to the Columbia wild type and ATGPXL5 TDN insertion line, it has lower vitality. So based on this, we can say ATGPXL5 only, not only role in ROS homeostasis, but is also maintained through the cell vitality. So after that, in the second part of experimental system, we investigate the involvement of GPXL5 in the development, rate of seed germination and growth development. So first, for that, we introduce the overexpressing lines, and based on the expression level, we choose two, overexpression 1 and 2, which is highly expressed. And these were the transformants. And we choose these two transformants lines for the further experiment. And the first experiment was like, do they have response in salt strength? And the first experiment I did with the seed germination. Under control condition, there were no differences. I found it. But when we apply the salt stress, so it's delayed this seed germination after 2 days in the Columbia and this ATGPXL5 mutant lines. But surprisingly, we found that very interesting. Like after 2 days treatment, they were not inhibited. So based on that, we can say like ATGPXL has a role in salt stress and providing protection to the seeds while it's germinating. So after that, we check the effect of this GPXL5 on root development under salt and under the salt and control condition. And if you see in the ATGPXL5 in the control condition, it was lower compared to the Columbia and overexpressing lines. You can see it's like significantly lower root length they had. So if you see with the graph root length, compared to the Columbia wild type and other overexpressing lines, it was lower. And also, if you see the fresh weight, it was only higher in the overexpressing lines. But yeah, after that, when they apply the salt stress condition, you can see, these parameters, root length. If you see the root length and the fresh weight, it's become, down. It's become down compared to the column to the control condition. But if you compare it with the Columbia and the mutant in the salt stress condition, they were less inhibited compared to this both. The same parameters were observed, like fresh weight was also more higher compared to this salt treated Columbia, and the mutants lines, it was higher. So after that, we checked the number of lateral roots, and we found nothing differences in number of lateral roots in the control condition, but the lateral root density was higher because it was calculated with the number of lateral roots divided by root length, and we found more lateral root density because of lower root length in the mutants plants. And after applying the salt stress, we found in overexpressing lines, they are responding very nicely, and less inhibition was observed compared to this Columbia wild type and ATGPX5 tDNA insertion line in both cases. So based on this, we can say this GPXL5 has a very important role in regulating the root length, fresh weight, fresh weight, and the lateral root development also. After that, we were very curious, like if they have a role in development, what could be in the shoot development. So this is the non-destructive method we have done, in the collaboration with the Professor Laszlo Sabados. Like we utilize the same approach which they have published in Frontier Plant Science before. And I utilized this plant size system, and we found like in the compared to, we check the fresh weight, rosette size, convex area, and convex percentage. And if you see in the mutants lines, they were lower compared to the others genotype. And after the applying of the salt 24 hours, we found that not 24 hours, it's like 10 days because it's seedlings, and we have seen like less reduction compared to this. It was observed in overexpressing lines, and convex percentage area was lower. So we can say this GPXL5 grew better in the presence of 100 millimolar NaCl. So with this plant size software, we were able to measure this chlorophyll content and anthocyanin content, and we also found the similar pattern as we studied in previous slides. And based on that, we can say, like if you see is lower chlorophyll and lower anthocyanin content in ATGPXL5 seedlings, but higher after applying the salt stress, they become more sensitive, but is less sensitive compared to this salt treated Columbia and wild type. So after that, the next target was like to check after the effect of GPXL5 in 6-week-old hydroponic plants. So first we check the expression level of ATGPXL5 in root and shoot with the help of qPCR analysis. We did it, and we found like in the control condition they were highly upregulated, and after NaCl they become reduced. But if you see the compared to the Columbia Col 0, they become 1/3. So we can say that ATGPXL5 is involved in salt stress. After that, so we would like to know like how they are involving in the ROS, and subsequently how they are helping in preventing the lipid peroxidation. So we check hydrogen peroxide content in the 6-week-old plants after 24 hours treatment, and also the MDA level, which is the indicator of lipid peroxidation. And we found that this hydrogen peroxide content in the root and shoot was higher in the ATGPXL5 mutant compared to the Columbia Col 0. And also when we apply the salt, we found only the higher hydrogen peroxide content in the shoot of Columbia Col 0 compared to the others. And also we observe if we observe this hydrogen peroxide content, elevation was more significantly higher, we reported in shoot compared to the if you see it's not it's higher, but it's not significantly, we didn't detect it. So after that, because ROS accumulation also induced the lipid peroxidation, but we found, if you see, is like MDA content was also higher in the root of mutants plants, but there were no differences we couldn't detected in the control condition. So based on these, we can say it's like 80 GPXL5 overexpressing roots and should had like mostly similar S2O2 and level S2O2 level. So I can say they are regulating more efficiently this ROS homeostasis. After that, we also check these two most important enzymes is also is enzymatic antioxidant enzymes, glutathione peroxidase and thiol peroxidase. And we didn't found any significant difference in root and shoot in respect of glutathione peroxidase and thyroxine peroxidase activity in the control condition, except this overexpressing length, which is poses very less, compared to other genotypes, is having lower amount of GPO activity. After salt stress, we found the higher glutathione peroxidase activity compared to the control condition, but we didn't detected any higher activity in thyrodoxine activities in the response of thyrodoxine peroxidase activity. So after that, there were lots of hydrogen peroxide or ROS processing enzymes. We also investigated, and we found like very alteration. We found alteration in antioxidant enzymes like SOD, APX, and catalase. So there are there was no constant trend, but more alteration we were detected. In mutants lines compared to the overexpressing lines, because they have the similar ROS processing activity like a wild type. So on the basis of hydrogen related enzymes, we can say it's this GPXL5 isoenzyme has a role in ROS processing as well. So after that, there are some GSS related antioxidant enzymes. So GST also is like thiol peroxidase, which has the similar activities like glutathione peroxidase, which use glutathione as a reducing agent, and it's reduce hydrogen peroxide in less things. So we also check this GST and GR activity, and we found GST activity only it was higher, it was higher only in overexpressing root, but we didn't find any differences in the shoot. But after applying the salt stress, the GST activity was upregulated in the Columbia wild type compared to the others. Also, we checked the glutathione reductor activity, and we didn't find any significant differences. They were elevated, but not significantly. But in the suit, they become lower compared to the Columbia Colgate. But after applying the glutathione reductase, applying the salt stress after 24 hours, we check this glutathione reductase activity in this mutants lines, and we found more GR activity if we compare to the control condition. So based on these, we can say this is having a role in the in a ROS processing process. So after that, based on this, now we explore the one side of this defense system like enzymatic, but now the turn is to investigate this non-enzymatic defense system. So in this non-enzymatic, we detected this ascorbate and glutathione, because they are the majorly which is involved in the non-enzymatic defense system. And we also investigated other things beside ascorbate and glutathione. We also detected proline, polyamines, and ethylene also, but I didn't mention here. So in this cases, like we check ascorbate content in the root, and we didn't find any significant differences under control condition. While we will applying 24 hours treatments, if you see, this is reduced ascorbate and oxidized ascorbate, both were elevated in mutant and wild type plants, but we didn't find differences. If you see here in the overexpressing line, we didn't find significant difference in the this reduced ascorbate level. But we found surprisingly the level of oxidized ascorbate was higher compared to this control condition. After that, we found some more alteration in glutathione content, and the lowest amount of glutathione was reported in 80 GPXL5 mutants compared to the other genotypes. After applying salt, we found like more oxidized glutathione were reported in the this 80 GPXL5 tDNA insertion line. And also, interestingly, we found like overexpressing lines contains more reduced level of glutathione content compared to the Columbia, and there were no significant, or it's become lower the level of this oxidized glutathione level compared to this. So after that, based on this, we use the nullness equation to quantify or to know the half cell reduction potential. So based on this reduce and oxidize glutathione, we calculated the redox potential of the root, and we found, because it's depend on the reduced glutathione level, it was higher the glutathione, so that's why it's become more negative, which is good for plants growing. But if you compare it, because this is the lower amount, if you see it here, is compare is become like more positive. It's towards minus. It's like minus 222, which is showing is a redox. It's a showing it's an oxidized state, but it's showing it's a like a reduction. Like it's. So we can say it's like GPXL5 actively participating in maintaining the redox state by maintaining through the reduced level of glutathione content in root. So after that, we check the level of ascorbate and glutathione in food, and similar, more or less similar alteration we were reported. And in the control condition, there were not so much differences. But if you see, there is no but higher amount of ascorbate, reduced ascorbate compared to the 80 GPXL5 was reported. But in oxidized one, there were no differences. We didn't report it. After applying salt, we found the Columbia Col Zero wild plants, they accumulated less ascorbate level. And after that, if you see in this mutants line, more like ascorbate, or I would say total ascorbate content was higher compared to the control condition. And if you see, is more accumulation was reported in the overexpressing lines compared to this Columbia, compared to this control condition, and we didn't find any differences in the oxidized ascorbate content. After that, more glutathione content was accumulated in the overexpressing lines compared to this wild type and this mutant lines. And when we applied the salt stress after 24 hours, we didn't see any differences here. But in overexpressing lines, if you see it, it's become lower. The level of oxidized glutathione become lower, and it was remains same here is if you compare to this control condition. Based on that, we calculated the redox potential, and we found it's like more redox potential we found in if you see compared to this Columbia or mutants plants, we found more negative. It's like more reduced form compared to others. So we can say yes, in root and shoot both it's actively participating in maintaining the redox status. So based on my, I would say it's like, I would like to summarize my lecture in 3-4 points. So I would go thoroughly with that. So ATGPX5 might play an important role in rose homeostasis and maintaining the cell's vitality under control as well as under salt stress. In the ATGPXL5 overexpression plants, antioxidant mechanism work at the level of wild type plants, but ATGPXL plants can be characterized with altered antioxidant mechanism, because in overexpression line, more or less they work at the same level of wild type. So these results confirm that ATGPS5 has function in regulating the redox state due to the elevated GSS content and lower level of GSSG content and more negative redox potential due to of higher level of GSH. ATGPXL5 enzyme. So based on this plant size software, we can say ATGPXL5 enzymes are required for healthy growth and development of Arabidopsis thaliana seedling both under control as well as salt stress condition. So I would say, I would conclude my lecture like altered redox status may be the key point in the mechanism of ATGPXL5 enzyme and can be a common factor in the stress response and development of plants. The differences found in the phenotypes and salt response of the ATGPXL5 mutants and the overexpressing plants suppose a complex interaction with plant hormones too. That's why we did also a hormone study with ethylene, and we already published that result too also, but I didn't mention here because due to of the time scarcity. And the results we already published in environmental experimental botany in 2 papers. And after that, I would like to thank you for my supervisor, Cesari Olan, for providing the opportunity to work there during my PhD stay. And also head of department, Professor Dr. Tilafir, and also my colleagues. And I would like to thanks to the collaborator, Professor Lasso Sabados, Gabarigo, and Attila Fair, and also from the university, Dr. Peter Poor and Agnes Sabesi. I would like to also extend my thanks to lab assistants who helped me, to arrange the things during my stay, during my PhD. So thank you so much.
Thank you so much. Thank you so much, Dr. Riazuddin. Now we are open for question and answer session. Anyone? Yes, who may like to ask the questions? We are open for question and answer session. Yes. Thank you so much, Riyaz sir, for beautifully enlightening us to know the role of Arabidopsis glutathione peroxidase like isoenzymes and their role in salt stress conditions. Now I would like to invite Professor Dr. Nitin Labane to propose vote of thanks.
Hello everyone. As a convener of today's session, I like to thank Dr. Riaz for taking out time and be with us. I think since nobody has any questions as such, it means that everybody has understood what you are trying to convey to the audience. So it was a good talk. Now, before I give the vote of thanks, we need to have some the participants to come up here on the stage and give their feedback. And after the feedback, I will propose my vote of thanks. Now I request all the dignitaries to be there on the stage. Dr. Laszlo, Dr. Ravi, please, madam, yeah, Vishal sir. Okay. Now I request anyone to come up on the dais and give the feedback, please. Thank you.
Hello, everyone. Good afternoon. I am Apurva Jha, an ex-student of this college and Department of Botany, recently graduated actually. The moment a colleague of mine told me that the department was organizing this symposium, and I went through the brochure and I saw that my mentor's mentor, I can call him my super mentor, Dr. Laszlo, was coming over to give us a talk. I jumped at a moment's notice to register extremely quickly. So all throughout this year, all of the conferences that we've been attending, you might have noticed that they have broadly been organized under this huge umbrella of sustainable development. But I feel that we cannot even reach anywhere near these sustainable development goals, especially the goal of food security, by the end of 2030, if we don't talk more about these extremely important topics like plant stress physiology and plant immunity against pathogenicity. So I would like to take this moment to thank these silent heroes that we have in form of these scientists who toil away in their labs and give equal amount of time and effort in order to help us, in order to help humanity achieve these goals. So thank you, Sir Laszlo, Sir Ravi, and Sir Riazuddin for taking a fraction of your time, coming over here and giving us this extremely enlightening bucket of knowledge. So thank you, sirs, and thank you, Department of Botany, for organizing this beautiful symposium. Thank you, everyone. Thank you.
Anyone wants to come up here on the dais?
My name is Shreya Upadhyay, and I am an ex-student of our esteemed botany department. Actually, this seminar have helped us all to gain insight about what is recently going on, and it is important to know about stress physiology, because as we know that population is growing, with that climate changes are occurring. So it is important for us to create such genetically modified organisms which can in turn help us to grow in future. And it is important to know about these topics, because this gave me an idea that what I can further pursue in future. This has ignited a lot of curiosity, and I would like to thank all the mentors for answering my questions. Thank you, everyone.
Did anyone? Anyone there?
I'll take this moment of appreciation and try to keep it as quick as possible. I'll say this with complete honesty. When Nitin sir firstly put it on the group that a symposium is being held, I was first thinking to myself and discussing with all my friends, should we go or should we not go? But then Nitin sir said that he'll personally check who has registered or not. I thought we have no option right now. But now I'll say that all of us will agree that we do not regret being here, because how rarely we get an opportunity to be with actually accomplished people and learn so many new things. When we just read our textbooks, we think, when we complete our portion, we think we've learned everything that there is to learn. But when all these new researches, these new experiments, we realize how little we managed to touch on these topics. There's so much more to learn. Now, I heard a few people say, in my friends, that it's a burden to learn new things, but we can look at it as an opportunity to move forward with how times are changing right now. I'd like to thank Professor Laszlo, Professor Ravi Gupta, and Professor Riazuddin, Nitin sir, Vishal sir, Asmita ma'am, Taufik sir, Sharif ma'am, Dinesh sir. Zareen ma'am is not here, and everyone, and all the participants as well, and everyone who actually listened to the lecture and asked questions. Because I think I might have slept for 5 minutes behind because I wasn't looking there. But of course, again, I'll end this with another thank you to everyone.
Is there anyone who still wants to express? Okay, I think this will be the last.
Very good afternoon to everyone. I am Disha Gandhi from MSc Part 1. And I would like to really express my gratitude towards the professors who have taken so much of time. And even in their explanation, they have gone into step-by-step details on the problems they faced, how they tackled the problems, and how certain methods were proven confirmation that how you decide between this method if there is a certain experiment you're performing and there are two particular methods you can follow. How do you identify which is the best method that will be applicable to the thing that you are studying about? And it is going to be very helpful for us when we do our own research in these things, and especially when they have gone into the specific things that are causing the problem. Like, we know that the climate is a problem, everyone knows that water is a problem, but going into the detail of it and where exactly the problem occurs actually will help us in solving the issue in the long run. And I'm very thankful to all our professors for organizing this, and for our esteemed professors who have come from abroad and given us their time, which is like, time is money. So thank you so much for giving us your time.
Thank you. Thank you very much, Disha. I think I will stop. I will not call anyone now. So kindly excuse me for that. I think the success of the symposium lies with so many participants want to express themselves that, okay, something they had got, which they could not have got in a regular due course of time, regular lectures and practice. They could not have got such a huge, I will say the huge is also, I'm finding it so small, to be very frank, because the knowledge, it cannot be comprehended. Okay, so I think I should start with my note of thanks. So thank you everyone. First, I like to thank Professor Laszlo Shavalos, who has come from Hungary and taken up time to see us all, and for his beautiful lecture. See that man, there is a number of things we should learn from him, man. Right? Studied in Hungary, went to Central America. Last week he was there in Argentina. Every time he's moving around the world, trying to collaborate with young people, young scientists, and trying to bring people together. Right? So that is the best part of us. So I think at this age also, he said so openly that he is trying to shift, trying to understand into new domain of epigenetics. Right? See, shift is important, but then somebody is trying to learn at this time. हम लोग को अभी समझ नहीं आया, मैं बूढ़ा हो गया, अभी मैं क्या पढ़ाऊंगा और मैं क्या सीखूंगा. But I think this is what I'm trying to learn from Laszlo. So Laszlo, thank you very much there's no words that can express my gratitude towards you. Secondly, we had Dr. Ravi Gupta, who has come from South Korea, and he also shared his insight, right, as rightly said by Disha, that his way of telling was like telling a story. How did he start it? What are the problems that incurred in his research? And how does the thing started to unfold bit by bit. So you don't think that you will get a Nobel Prize in one day. I think I think Laszlo, in the very first lecture, very first slide, he showed that the Catalan who got a Nobel Prize this year, right? So I think that this shows it was a huge work, right? So I think I like to thank Dr. Ravi Gupta, and also thank Mrs. Gupta also, because she allowed Ravi to be here with us in Bombay. Right? So thank you, Ravi. I like to thank Dr. Riaz Riazuddin, who is there doing his postdoc in Spain. In spite of his busy schedule because he joined this new lab very recently, and we can understand to set in a new lab and to be there up to their expectation, it needs a lot of hard work. And in spite of that, on my request, he agreed that okay, he will be there for this particular international symposium. So from the bottom of my heart, I like to thank Dr. Riazuddin for being with us. Now, these are the most important people, but you all are also very important, because without you participant, this symposium could not have been successful, right? So I like to thank all the participants who has come from different universities, institutes, different firms, who had taken out some time, because that shows at core you are a botanist. At core you had love for the plants, and because of that you are here. So I like to thank all of them. The conference, the symposium was organized for the students, and I think we need to clap for the students, because I think the feedback that has come from the student, it tells that they were able to get different insights. So thank you all student participants from this particular college and from the different colleges who has come out here, the UG as well as PG and the PhD students out here. Principal Madam, especially all the staff members of Botany Department. Yeah, Botany Department and other departments, different HODs, different faculty members from Bhavan's College and other colleges who has also come, and they had been here throughout. So I like to thank all of them. Special thank for Mr. Sandesh. Because of Sandesh, Nitin Patel, then Ramesh Paloti, because this whole function, even some of you those who might have missed, your friends who might have missed, the whole function has been recorded and it is on Google Live, YouTube Live, it is there. So those people who couldn't understand at the first glance, you can very well go back to, it says on Bhavan's YouTube channel. So you can very well go back to that channel, listen to it, because it might be difficult, because we can understand, because this is all genetics, proteinomics, everything is there. So it will be difficult, but when you listen it 2 times, 3 times, I think you will get much more better perspective of what these legends has been talking about. Okay, so I like to thank these people. Also, all those who directly or indirectly helped this international symposium a great success. If I missed someone, so please excuse me. So once and all, I like to thank everyone. I think Laszlo is saying that he wanted to say something.
Just a small note that you mentioned Kariko. You mentioned Kariko Catalin. I know her for 50 years because we were in the same group in the university. So it was nobody expected that what will happen with her. But now there is a book of autobiography that is appeared in Hungarian and English. And I think I would recommend, I think it will be in India available also. So I'm not sure how is the policy with this book printing and selling. But I encourage people to read that book, because this is an example that has how a young girl from a very modest family, she was really coming from, I visited her family, revisiting, so was very stubborn. Well, she's also very talented, but she was also very stubborn to work, repeat the experiments, not once, not twice, but I don't know how many times with all the controls. And she was very exigent for herself. And this biography shows also that the way that she pursued her research was not a very smooth thing. She was faced by problems in the US. She was faced by sometimes by problems with her pulse, and then she find a way to go on and to pursue her ideas. And which is very important, she was very exigent. So until she went to BioNTech company, she was making all experiments herself. She had no staff of 20 postdocs, 30 students, and such. She was standing in the lab, making the experiment, sometimes 3 or 4 o'clock in the night, sending a message to his colleague that, okay, I find things, and then 10 minutes later, she got back the answer that, it's fine, go on or correct this. So time is sometimes is relative. So it's not that, okay, lunchtime, I threw away my pipette and come back next day. So this is the way how she was making the science. And I think this book would be a nice read for those who have their own idea, especially for women, because I see that the most of the audience is here in women, that even if you come from a modest family, but you have a talent, you have a diligence, you can get really nice experiments and nice experience in science world.
Thank you. He was talking about Catalin, who got the Nobel Prize this year, right? So I think we always try to walk down the trails, right? That is the reason why we read autobiographies. So he was telling us that let's read the autobiography of a person who got a Nobel Prize. So we'll understand if you can also walk down that trail, there's every chance that you will also win a Nobel Prize. And we want many of the Indians to win the Nobel Prize. Yes. Yes. Any energy nahi hai kya? You don't want it? Yes. Okay. Thank you very much. Now we'll. I want the guests to be here, and I want the volunteers to be on the back side so that they can have the group photograph. After that, we will have the photograph of everyone up here. Please, volunteers, please. आ जाओ, पीछे में खड़े आ जाओ. I declare the function is formally over. Now we are just waiting for the. And after that, we'll have a national anthem. Or first we'll have national. Okay, let's have a national anthem first, and then we'll go wait for the photography. Then we'll officially end. I request everyone to stand for the national anthem. Yeah. जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात द्राविड़ उत्कल विंद हिमाचल यमुना गंगा उच्चल जलहित तव शुभ नामे जागे तव शुभ आशीष मांगे गाए तव जय भारत गण मंगल नायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे. Thank you very much. Now for the let's come for the Sandesh, please हे बंद करून टाका, ऑनलाईन लाईव्ह बंद करून टाका. लवकर लवकर पटकन. Staff members from the please. Staff member.
पुढे बॅनर आहे ना.